Okay, so we're going to get started with the afternoon session, and first want to acknowledge our gold sponsor here from BioLegend. Would you like to say something?
Thank you for having me. It is just a pleasure to be here. When I initially got the invitation from Safran, I told my colleagues we have to come. I'm really excited. This is the first symposium that we're sponsoring here, and I hope that it's just many more to come. So thank you for allowing me to take part in this event with you.
Thank you. Our next speaker is Dr. Jennifer Condon, an associate professor here at the CS Mott Center for Human Growth and Development at the Department of Obstetrics and Gynecology at Wayne State University. And the talk is on resolving inflammation during pregnancy.
Hey, everybody. Thanks so much for attending today. Hope you all aren't too sleepy after your heavy lunch there. So I work on pregnancy and preterm birth, and really trying to define why women go into labor at 9 months. Why doesn't it happen earlier? Why is it only 10% of pregnant women go into preterm birth? What are those regulating factors that keep a pregnancy intact until 9 months? And it's kind of startling that we don't know why a woman goes into labor at 9 months. We know the baby is ready, but we don't know the molecular mechanisms that trigger that laboring phenotype. So because of that, okay. Because of that, particularly in Detroit, we have a really high rate of preterm birth, 15.1%. We're the worst urban population for preterm birth in the US. Again, this is quite startling considering how good we are at treating babies once they've come out, but we're not very good at keeping them in. And as we heard earlier, there's really no successful treatments for preterm birth. A lot of them are complicated, and our hero progesterone is now also off the market. We have drugs that we can use to stop the uterus from contracting for a short period of time, so we can throw in some corticoids in there to help it develop the baby's lungs. But other than that, we're stuck. So any of the tocolytics that we use basically block for a short period of time. So what we do know about labor at term is largely inflammatory. So inflammation drives the production of prostaglandins locally within the uterus, and that causes the uterus to contract. Preterm labor is the same. Preterm labor, we get inflammation causing prostaglandins to be produced, and that causes the uterus to contract. Preterm labor is largely driven in 60% of the women by infection. So infection is driving that inflammation. The women we're really interested in is the women who have this sterile inflammation. They don't have an infection, but they trigger this same inflammatory reaction and causing uterine contractions. And for us to understand what actually happens in term and preterm birth, we believe that we have to know what's happening in a normal pregnancy. And how is a normal pregnancy, how is inflammation limited across gestation? Why are all of these women not undergoing premature uterine contractions? What is limiting that from happening in a normal pregnancy? So this is just looking at the inflammation profile during a normal pregnancy. Early in pregnancy, it's very inflammatory. We have inflammatory cytokines being produced during the implantation period. You require inflammation to be there to allow implantation and embryo attachment. And then you can see in the mid-pregnancy, we have placental development and maintenance of pregnancy through the second and third trimester. It's largely anti-inflammatory. We have anti-inflammatory type 2 macrophages. We have anti-inflammatory cytokines being produced. And then again, at the end of pregnancy, it switches to this inflammatory phenotype, and you require that inflammation to cause the uterus to contract. And what we're really interested in is, why do we have this pattern? What is limiting that inflammation early in throughout pregnancy? And this is, again, it's reiterating that you have this large inflammatory profile during implantation that declines and then returns at term with this not anti-inflammatory, but inflammation being resolved there in that second and early third trimester. Okay, so we know during a pregnancy that every uterus in every woman is exposed to a huge amount of stressors. So you have, and a lot of these are rat and mouse models, this is human, but from changes in hormones, changes in cytokine profiles, changes in mechanical stretch, hypoxia. Early in pregnancy, the uterus is completely hypoxic. That changes over time with vascularization. So all of these events, if they go wrong, can cause massive inflammation. So why don't they? And what we've been working on is really looking at this response, the unfolded protein response. It's a response that occurs within all of our cells, within the endoplasmic reticulum of your cell. And it basically is the mechanism that allows you to adapt to a stress. And all of our cells do this all of the time. It's a very normal response. It is not a bad thing, it's a very good thing. You see a stress, you adapt to that stress and overcome it. You eat too much glucose, like you've done at lunchtime, your pancreas will produce more insulin, for example. This is the mechanism that makes more insulin and ships it to where it needs to be. So it helps you go about your day. So this is this process. And when you have a stress, like too much insulin, or we use glucose starvation in our model, you can actually limit the amount, not limit the amount of proteins being folded, but you can kind of overextend the ability of the proteins to be folded correctly. So you get an accumulation of misfolded proteins. You don't ever want that. You don't want misfolded proteins being made and transported out of your cells. So you activate this adaptive unfolded protein response. This largely means that you have upregulated the chaperone proteins that help with protein folding. It comes into your misfolded proteins, fold them correctly, basically upregulates the ability of the endoplasmic reticulum to fold proteins correctly. And in doing so, it will allow homeostasis to be regained, which is really required for efficient protein folding. The misfolded proteins go away and everything goes on as normal until we see the next stress. However, if you have excessive stress or prolonged stress, you will activate a different pathway. If the capacity of the endoplasmic reticulum has been overwhelmed and you can't get rid of those misfolded proteins, you'll activate this pathway, the CHOP pathway, that activates apoptosis, inflammation, and cell death. And that basically gets rid of a cell that's misfolded proteins, that can't fold proteins correctly anymore. And it eliminates that cell. Okay, so what we have found, a link between these two pathways. So what we're saying is that the uterine stressors that every pregnancy sees basically utilizes that adaptive process to maintain a pregnancy. And it allows you to adapt to all of the stresses that you see during a pregnancy. And in our proposal, what I'm going to talk to you today about, it really works to limit inflammation during those stressful events. And that is the way uterine quiescence is maintained across a normal pregnancy. What we found also was that when the uterus has been activated to promote this adaptive signaling pathway, it starts to secrete very specific proteins. And we can find these proteins in the blood of pregnant women. And it allows us to define whether a woman has adapted correctly to a stress or not, and whether she's at risk for preterm birth or not. So we stress uterine myocytes, this is in culture, using a process called SILAC, where you heavy label and light label your cells. And then what they secrete into the media is now identifiable by mass spec in response to stress. So one of the biggest proteins that we found that was upregulated is HSPA5. It was almost 25-fold increase being secreted under a stressed condition. HSPA5 is also called BIP. It's also called GRP78. For the rest of the talk, I'll call it BIP. So what BIP normally does, BIP is this chaperone protein that we just talked about earlier inside the cell. It helps fold proteins. It helps you accommodate those stresses. But what we've found is that it can also be secreted out of the cell. And this is known. We know that BIP actually comes out of the cell. It's not because the cell is undergoing apoptosis. It's a normal process. It can escape the cell. And here is a tissue culture showing that if we hit the cell with clinical, which is a stress, we can upregulate the levels of BIP inside the cell. And then this is a ELISA showing if we take media from around these cells, BIP has been transported out of the cell into the extracellular compartment, and it's readily identifiable. Okay, so now if we look across gestation, this is in a pregnant mouse model, we see that BIP corresponds nicely to when we have this anti-inflammatory portion of a pregnancy. It's low with implantation, it's gone with the onset of labor, suggesting that possibly this BIP plays an important role in regulating that reduction of inflammation in this second and third trimester, and that the absence of it would trigger an inflammatory response. So we tested this hypothesis. We overexpressed BIP GRP78 in uterine myocytes, use a doxycycline-inducible lentivirus, and here is 99% of the cells were overexpressing GRP78. Here is its overexpression, we're over 40-fold overexpression. Western blot showing its elevation. And what we found was that when GRP78 was up, intracellular, we could actually limit inflammation. So IL-6 levels are decreased. So just endogenous IL-6 levels are being ablated by elevated levels of BIP. If we do the opposite, if we use siRNA to knock down BIP, so here you see across here we've knocked out BIP. When we knock it out, you see there's massive induction of IL-6. So without BIP, you have massive inflammation. With BIP, you don't have much inflammation. And actually, this was Western blotting showing IL-6, and then this is the secreted cytokine in the medium. This is an experiment that we did again on cells. We did it in placental cells. We also did this in uterine cells. And here we're treating cells with LPS. Lipopolysaccharide mimics a bacterial infection. And this is why most women will go into labor, triggers an inflammatory response. So here we hit the cells with LPS, we get a beautiful induction of NF-kappa B, which is a marker of inflammation. If we hit the cells with BIP and LPS, we see that inflammatory response has been ablated. We also see the same for IL-6, an inflammatory cytokine. Hit it with LPS, a beautiful induction of IL-6. Hit it with BIP plus LPS, that IL-6 levels are rapidly decreased, very significantly decreased. So again, the presence of BIP is actually resolving that inflammatory response that's been triggered by LPS, which represents a bacterial infection. And this was all done in vitro. What we've also shown is that we know that we can differentially polarize macrophages into an M1 type macrophage, which is inflammatory, or an M2 type macrophage, which is anti-inflammatory. And when we do that in the presence of BIP, we find that M1 type macrophage polarization is inhibited. So this is a normal M1 type macrophage polarization. In the presence of BIP, it doesn't happen. And if we just expose non-differentiated macrophage cells, just monocytes to BIP, we see that they automatically go to an M2 type macrophage, which is a pro-resolving anti-inflammatory macrophage. So again, BIP not only can reduce the cytokines that are being produced and limit bacterial-induced inflammation, it can also play a critical role in regulating inflammation by resolving it through the upregulation of these pro-resolving M2 type anti-inflammatory macrophages. So what happens in the human is it available? Can we see it in the circulation? And it is. It's very high in pregnant women. So this is a non-pregnant woman. This is a pregnant woman in her second trimester. We see it very identifiable and increasing with pregnancy. But what's interesting about this is pregnant women. These are 10 pregnant women in blue, 10 pregnant women in red, and looking at their gestational profile of BIP in their circulation. And the women in blue are the women who delivered at term. The women in red are the women who had preterm uterine contractions. And as you can just see when you look at them, when we pool them all together, the women who have premature uterine contractions had less BIP than the women who had normal term delivery. This would suggest that if you have lower levels of BIP, you're more likely to have an inflammatory response, or you're more likely to have a uterine contraction. So it's kind of following our hypothesis. These are women that, so this is a non-pregnant group, normal pregnancy, a pool sample from 20 to 37 weeks. This is women with term labor, and this is women with preterm labor. Now these are women, these are the idiopathic preterm births, the women who do not have an infection, but have that nice inflammatory response that's not caused by a bacterial infection. And as you can see, they have much lower levels of BIP in their circulation in comparison to term laboring women. Again, suggesting that if you have lower levels of BIP, you're more likely to have an inflammatory response, you're more likely to go into preterm birth. When we look at our data another way, what we can see is that the serum levels of BIP, even in the first trimester, can predict whether or not you will go into preterm birth with 81% accuracy. And of course, that gets nice towards the third trimester, we have a 91% accuracy, but we really want to target these women early in their pregnancy. Are your levels low enough? Can we intervene? Can we change how you're going to respond throughout your pregnancy? Can we prevent you from going into preterm birth? And that's where we're going with this. So we wanted to have a look in the mouse model to see whether or not we could prove it. Could we give them BIP? Could we change their gestational profile? And all of this work actually has been done by Melissa Bukowski. She's my graduate student, and she's done an amazing job. These are hard experiments to do. This is a preterm birth mouse model at gestation day 17. We hit them with LPS, which always induces preterm birth. But prior to that, 3 hours prior to it, we gave them BIP. Then we hit them with LPS plus BIP at the same time. And then an hour later, we gave them BIP again. So they got 3 doses of BIP and 1 dose of LPS. And this is a very long experiment. We had to try and figure out the dosages and the time, the day to deliver, and the timeline of delivery. So this was hundreds of animals. But what we found was that LPS induces LPS induces preterm birth, and 25% of the animals are still alive in this preterm birth phenotype. If we give them BIP, we actually see up to 65% of the animals are alive and being delivered at term. BIP alone has no effect versus control. So we're actually able to reverse the preterm birth phenotype induced by a bacterial infection by pre-dosing and co-dosing with BIP. So what does that look like? These are LPS treated animals. They're all the same gestation stage, taken at 18.5. This is LPS. This is the LPS plus BIP group, and this is BIP alone. And I don't have a control of the dose, sorry. But as you can see, these animals are fine, they're alive. Their weights are no different from the controls. But as you can see, BIP in the presence of LPS is able to replicate that preterm birth dead phenotype that we see with the LPS. And then just looking at the molecular side of it, this is looking at the uterus of mice treated with LPS and BIP. And one of the things that we've noticed prior is that if you treat a pregnant animal with LPS, the endometrium becomes apoptotic and the placenta detaches. So we use that as a marker for the ability of BIP to limit some of those detrimental effects that are occurring with LPS. So these are just different magnifications in the uterus. LPS has beautiful apoptosis. This is tunnel staining, fluorescent green in the endometrial compartment only. Whereas when we treat with BIP plus LPS, we see very little or no tunnel staining, suggesting that BIP is able to rescue that apoptotic phenotype that you see in the endometrial compartment when treated with LPS. We also see cytokines like IL-10, these pro-resolving cytokines, are upregulated in the BIP group, and this reaches significance versus control and LPS. Remarkably, what we see in the LPS group is a massive induction of inflammation. Seen here, this is a Western blot for TNF-alpha, and we see that that's absolutely ablated in the animals that were treated with BIP and LPS. And again, BIP itself, control cells are at baseline. So again, we're showing that BIP is able to resolve that LPS-induced inflammation in vivo in pregnant animals. These are the uteri of, this is like 3 individual pregnant moms. So it's very significant. So the conclusion would be that we believe that the pregnant uterus across gestation experiences a whole pile of stressors that can cause inflammation and lead to premature uterine contractions. And to avoid that throughout pregnancy, we use the unfolded protein response. And activation of that unfolded protein response then allows the release of BIP intracellularly and then extracellularly. And then that extracellular BIP throughout your pregnancy resolves the inflammation that you're seeing. It's like a break all of the time, just breaking on that. When BIP disappears, then you get a nice spike in inflammation that causes your uterus to contract. So where do we go with this? We see that this is just a recap. Extracellular BIP can promote the resolution of inflammation through macrophage polarization and circulating PDMCs. We see them changing. So what we think is that idiopathic preterm birth that's occurring independent of infection and infection-induced inflammation is really likely driven in pregnant women by this unbalanced unfolded protein response. There's women out there that don't have the capacity to adapt correctly. So if you have less of this BIP, less of this unfolded protein response, then you're more likely to have an inflammatory response and go into preterm birth. So then we do think that maybe we can use this not only as a biomarker for identifying women who are likely to go into preterm birth, but we also think, can we just use it as a drug? Can we top up these women that we see in their first trimester that have very low levels of BIP? Can we just can we just prop up their levels of BIP by increasing systemic levels? Will that be enough to actually suppress the inflammatory responses that they're incurring due to not having enough? And will it just restore the appropriate inflammatory profiles that allows resolution of that inflammatory response? And then that would prolong uterine quiescence. And then we also do think this has led us to a novel opportunity for preterm birth associated biomarkers. And that's it. So Melissa is the graduate student that did most of this work. My husband, Eduardo, also works here, and he's been my longtime collaborator. Gail has been fantastic, and Rick Pilsner we've done a lot of work with. Christina Adams-Walsdorf, we've done a baboon study with her that I haven't gone over. The human study, the samples came from Roberto Romero, and Addie has been sending me all of the data from that, which is great. And then we've been funded by EMAG, Marta Dines, a very important CS Mott Center pilot grant has helped us on our work. So thank you. Yes.
Fantastic talk. Thank you. I think BIP can actually be a potential target for inflammation. My question goes to, can BIP distinguish between what I call good and bad inflammation? Because sometimes, even though preterm birth can be bad, sometimes we would prefer the patient to deliver preterm if they have a bad infection.
Right.
Because if you stop the infection, they get, it could kill them quickly.
So that's why we're actually really interested in the idiopathic preterm birth group. Like if a woman has an infection, we don't want to keep an infected amniotic cavity within the woman. We want her to deliver that baby preterm. So we would not ever give BIP to a woman like that. I guess just doing an amniotic profile along with a serum profile to see whether IL-6 levels were up in the amniotic fluid would be able to, prior to treating the women with BIP, we would be able to categorize them correctly so that we weren't doing that, right? That's a great question.
So Jennifer, I might have missed it. What's the mechanism of action? Does BIP bind to anything?
So I didn't have time to go into all of that, but it's actually through the toll-like receptor. So we're seeing BIP in collaboration with another protein is actually able to bind to the toll-like receptor and endocytose it. So you're taking toll-like receptors. Toll-like receptors help and trigger inflammation. So like LPS will bind to the toll-like receptor and trigger an inflammatory response. So BIP now binds to the toll-like receptor and takes the toll-like receptor away from the surface of the cell. So now it doesn't matter how much LPS you have out there, you're not going to trigger an inflammatory response if you don't have the toll-like receptor out there. And that's very specific. BIP seems to be doing that very specifically.
So is it TL?
TL seems to be 4 along.
And is it incorporated into endocytotic vesicles or something like that?
It is, and then it's degraded. But it's actually short term. there's like a 3-hour window. Actually, in the mouse studies, we actually see changes in TLR4 also in vivo, so it mimics what we see in vitro. But that's why we chose the 3-hour time point, because to pre-treat the mice with BIP, because it takes 3 hours for BIP to get through the system to hit the toll-like receptor and then to take it away. So that's why that was the timing issue that we had with the mice, trying to design that.
So did you move longitudinally and ever deep? So women that don't have enough of it, is it not enough at the beginning that stays there like that, or it fails to raise enough, or what happens?
So if you look at the human, profile of BIP, it's very broad, right? You know, there's very little BIP in some women that go into term birth, and because the sample sizes were so huge, you could get significance. When we did the longitudinal studies of the women who had term labor and who had preterm labor, we were trying to identify the beautiful profile that we see in the pregnant mouse. And we couldn't identify that profile at all. But when we did the study in the baboon, what we found was that BIP in the baboon is cyclical. It comes up and comes down all of the time. So the fact that we could even get significance in the human studies with all of that range would suggest that if we could take all of those women at exactly the same time point, or give them a stress to identify whether they were responding correctly, and everything was collected at exactly the same time under the same conditions, then we would probably have we would probably be able to identify the same profile that we see in the pregnant mouse.
Serum and plasma would carry the same information for BIP, or only work in serum?
Yeah, we have checked in plasma too. It's the same. We're getting the same. Yeah. And we're just about to start on blood spots to see if we can do ELISAs with those too.
Thank you.
Here's my question.
Sorry, is it involved in other inflammatory diseases?
So, several. So, collagen-induced arthritis. If you inject BIP into the arthritic elbow or ankle, I can't remember what it was, they could actually resolve inflammation that way. If somebody has somebody has lung cancer, for example, and you just take their blood, they'll have elevated levels of BIP in there. So it's actually a marker of general well-being, basically. Thank you.
Thank you, Dr. Condon. The next talk is by Dr. Marta Rodriguez-Garcia, Associate Professor at the CS Mott Center for Human Growth and Development and the Department of Biochemistry at the Wayne State University School of Medicine. And the title of the seminar is Understanding HIV Infection in the Female Reproductive Tract.
Thank you. Thank you so much. I'm very excited to be presenting here. We recently moved the lab to the Mott Center, so I'm very glad to be able to present our work to this audience. I'm going to change gears a little bit. We don't do pregnancy. We're very interested in infections of the reproductive tract, and in particular in HIV infection. And the goal of our lab is to understand how HIV infection is established in the female reproductive tract. So since HIV was discovered in the 1980s, more than 60 million people have been infected around the world. And currently there are about 40 million people living with HIV. Of those, half of them are women. And in the endemic areas like sub-Saharan Africa, more than half of the people living with HIV are women. There is an age group that is very important for research, which are young women and girls. And they're known to have enhanced risk for HIV infection, particularly in endemic areas like sub-Saharan Africa, where young women are 6 times more likely to be infected by HIV than young men. So a lot of research goes into this young group. And importantly, women acquire HIV through sexual transmission. So if we want to develop preventive strategies, we need to understand how infection is acquired and established. But HIV is not only a problem for young women. And if you look at data from the CDC in the US, women have HIV throughout all the ages. And this is not only to the fact that they acquire HIV when they're younger, and now with the antiretroviral therapies, they live longer in life, but there are new infections happening in every age group. So if we want to prevent HIV in all women, we need to understand how the immune system works in the female reproductive tract and how it changes as women age and how the immune response is changing as women age. So the current model for HIV acquisition, this is a representation of the female reproductive tract with the lower tract with a stratified epithelium represented here, and the upper tract with a single layer epithelium represented here. Underneath and in between the epithelial cells, we have all the immune cells that will be target for the virus, but also responsible for protecting against the viral infection. So what we know from studies from many people is that as HIV comes in, it has to overcome the epithelial barrier, and then it will infect the HIV target cells. The main target cells in the female genital tract are CD4 T cells, and work from us and others have demonstrated that these cells are a specific subset, which is called T17 cells. They can also be captured by dendritic cells, which grab the virus and then can migrate to lymphatic tissues and disseminate the disease systemically. And as I was mentioning, these Th17 cells are the main targets in the lower tract, but they're not the main targets in the upper tract. And this is a link between the research I do and that I think will be of interest to the audience here, is that the same immune cells that we study for HIV infection and for genital infections are the ones that are going to be involved in pregnancy and reproduction. So for example, Th17 cells have been described to be bad for successful pregnancy. And we see that prior to pregnancy, they are very low in the endometrium. So the results and the responses that I study can be very relevant also for successful pregnancy. But something I didn't tell you is that the majority of women, when they are exposed, do not get infected. And that is information coming from epidemiological studies that demonstrate that about 1 in 1,000 exposures lead to infection. That means that 999 times that a woman is exposed will not get infected. And that indicates that there are potent natural barriers, potent innate mechanisms that protect women from infection. So that led our attention to the neutrophils. So neutrophils are abundant in the female reproductive tract, and they're known to play key roles in protection against bacterial infections. But the role in HIV infection is completely unknown. And we study innate mechanisms of protection against HIV infection. That's the main focus of the lab. And for the purpose of today's talk, I will focus on the work we've been doing with neutrophils. So I'm going to show you some published data looking at the role of genital neutrophils in preventing HIV infection. Some mechanistic work of how blood and genital neutrophils recognize and respond to HIV, and some studies we are doing now characterizing the tissue distribution of the neutrophils and the transcriptional profiles of these genital neutrophils. So to do our studies, we obtain human hysterectomies, and we study the endometrium, the endocervix, and the ectocervix. These are tissues from women that are HIV negative. In vitro, we digest these tissues and we release the immune cells. And then we perform in vitro studies where we can purify specific cell populations and perform functional assays and HIV infections in vitro. And we do it this way because that way we can investigate the early responses, the responses that cannot be investigated in humans because women get infected and they don't know when they're getting infected. And there is no way to in vivo study the early events that happen in the first minutes or the first hours after viral exposure. So this study is actually the interest in neutrophils studied by chance when we were looking at other cells, the dendritic cells. So to do that, we were taking the mixed cell suspension. I'm gonna go back here to show you when we digest the tissue, we obtain what we call the mixed cell suspension, which contains all the immune cells and fibroblasts. So using that, we added HIV viral particles, and then we looked at which cells capture the virus. And we published these studies a while back. We saw that the dendritic cells capture the virus, and then we did the opposite exercise and said, okay, what other cells can capture the virus? And according to the literature, we were expecting to see macrophages, but it was a surprise when we saw that it was actually the neutrophils, the ones that were capturing the virus. And we did confocal microscopy to look at the neutrophils, but instead of finding individual cells with virus in them, we found these big clouds of DNA that were completely coated with viral particles. And this was a surprise. I thought, could this be neutrophil extracellular traps? So neutrophil extracellular traps are fragments of DNA that are expelled outside of the cell, of the neutrophil, when it is stimulated. And it's been described as a protective mechanism against bacterial and fungal pathogens. But the role in viral infections is not well defined, or it was not well defined at the time this study started. And this DNA is coated with lots of proteins that have broad spectrum antimicrobial activity, including anti-HIV activity. And when I had this observation, I went into the literature and I could find one paper that had described that neutrophils from blood can trap HIV in nets. And I want to emphasize the difference in the significance of the finding, because if you have HIV in blood, that means that person is already infected. So the nets can be important for disease progression. But if you have nets in the female genital tract, and those nets are able to capture the virus and inactivate the virus before it reaches the target cells, then we're talking about a mechanism for prevention of HIV acquisition. So we went back to the lab to investigate this in more detail, and we performed some studies to characterize and visualize the nets. And this is how nets look like. They're big, long fibers of DNA that are completely coated with proteins, as I mentioned. And here we have an example with myeloperoxidase, which completely coats the DNA. So when we do the same assay, but stimulating the neutrophils with HIV, we use HIV particles that are labeled with GFP. And then you can see each little dot in this image is an HIV particle that is trapped into these nets. So the nets can trap and immobilize HIV. But net release is a dynamic process. And we really wanted to capture how early and how this process is happening in real time. So we developed this assay to look at it using microscopy, in which we plate the neutrophils in the presence of a nucleic acid dye that is impermeable to the cell. So if the membrane of the cell is intact, the dye will not stain the nuclei. But if the membrane is compromised, or if the DNA comes out of the cell, then we can detect this signal. And in addition, we stimulate the neutrophils with our GFP labeled viral particles. And this is an example of a net that already happened. So here you can see the DNA signal. Here you can see the virus trapped into that net. And here is the merge. And these are videos. So you will see how another net is going to appear here. At the same time, we will see how that net is capturing all the virus. And here in the merge, you can see both of them happening at the same time. And when both signals are together, we can see yellow. And I like showing this image for multiple reasons. One of them is we have some dead cells in this image here, which are not HIV positive. The other one is we have a number of neutrophils around that are interacting with the nets, and yet they're not releasing nets. So that means there is a control. There are control mechanisms regulating this. This is not just random release of nets. So we developed this system with purified neutrophils, but we wanted to know with the regular proportion, the natural proportion of neutrophils we have in the tissues, can we actually detect this phenomenon? So this is using the mixed cell suspension with all the cells in the tissue. And, and this is a video that is not working. Now, so you can see very quickly, as soon as we add the virus, we see lots of nets appearing around. And as a point of comparison, here is the control. And you can see we have some dead cells here, but the image is completely different. And HIV induces a strong response for the release of nets. So we have a way to quantify this. We can quantify the red signal, which is the DNA. We can quantify the green signal, which is the virus. And then we can quantify both of them together, with which we call capture. And we use a couple of controls, one of them where we don't add the virus, and another one where we add the virus in the presence of DNAs, which is gonna degrade all the DNA to demonstrate these are in fact nets and not something else. And then we can quantify this over time. So these black lines represent the signal in the presence of HIV, and at the bottom we have the controls overlapping. And we did this with multiple tissues with different women, and we see that throughout the female reproductive tract, neutrophils release nets in response to HIV. But the big question is, do these nets inactivate HIV? Are they preventing infection? So to answer that question, we developed a protocol to purify the nets. So we purified neutrophils and stimulated them to release nets, purified the nets, and incubated the nets with the virus, and then added this to CD4 T cells, activated T cells that serve as target cells for the viral infection. And this virus that we're using here is a reporter virus that when there is infection, we will see green. So in this case, these are clusters of infected cells that you're seeing here, lots of green signal indicating there is infection. But if we have nets present in the culture, there is nothing. And we did this with multiple patients, and we quantified the percent inhibition of infection. And by day 7, we have approximately 100% inhibition. And this is 7 days because this is the very well-established assay to evaluate infection in vitro. So nets inactivate HIV and prevent infection of target cells. And I started this talk talking about aging and how it's important to study protective mechanisms in women of all ages. So when we performed these studies, we had this observation that when we were using neutrophils from premenopausal women, we got a very nice, strong signal for net release. But when we got neutrophils from postmenopausal women, we saw lower signal of the nets. And actually, when we quantified it over time, women responded immediately to, the premenopausal women responded immediately to the virus, but postmenopausal women seem to have a delay in the response. And we quantified this with multiple women, and we saw that this delay was significant, and there was a difference in the initial steps of the net release, but not later on. And we did this with blood neutrophils and with genital neutrophils. And because we saw this difference between premenopausal women and postmenopausal women, we wanted to know if this was a consequence of age or menopause. So we followed them over age. We did a correlation with age and the area of the nets. And we saw that there was no correlation in the premenopausal group. But after menopause, as women age, there was a significant decrease in net release as they got older. So what is the mechanism? One of the mechanisms that is known for cells to recognize HIV is the toll-like receptors that were just mentioned in the prior talk. So we know that toll-like receptors can sense HIV, and we know that toll-like receptors can trigger release of nets. So the first thing we tried was blocking this toll-like receptor pathway to see if we could prevent or diminish the production of nets. So you're gonna see two videos here. In the first one is just HIV, and the second one is HIV with inhibition of these toll-like signaling pathway. And you can see in the HIV, you immediately see the nets being produced, while in the toll-like inhibition pathway, there is a delay. And we quantify this, and we saw that, in fact, when we block this toll-like receptor pathway, we have a decrease in net release, and specifically a decrease in that early stages of net release. And then we compare blood neutrophils, and we got a surprise compared to the genital neutrophils. Because when we compare the premenopausal women and the postmenopausal women, we did see this decrease in the postmenopausal women, but no effect in the premenopausal women. And that indicated to us that there were alternative mechanisms in addition to the toll-like receptors. Also indicated to us that the neutrophils from the older women were responding differently than the neutrophils from the younger women. So we went on to investigate alternative mechanisms. And one of the mechanisms that's been described in the literature is calcium. So calcium release is known to be involved in the triggering of net release in response to bacteria. So we investigated whether this was also true for HIV. To do that, we use a reagent that will become fluorescent when it binds to calcium. And again, you will see a video here. Here is the control with no HIV. Here is the HIV. And as we follow it over time, you can see some of the nets appearing, and you can see the difference in the signal for the calcium here, very strong compared to the control. And we quantified this signal, and we saw that this is the control here. And when we add HIV, there is an increase in calcium. And the surprise was that when we, or not surprising, in line with our results from the previous slide, in the younger women, we saw this very strong calcium response, but in the older women, this response was abolished. So we think this was this additional alternative mechanism that young women have, and as they age, is getting dysregulated. The surprise came when we looked at genital neutrophils, because we thought, okay, this mechanism is probably also contributing in the genital tract. But if you look here, the two curves are very, very close. There's minimal calcium release. And we did it with multiple patients, and we detected no differences between the control and the HIV. And this is evidence, to my knowledge, one of the first evidence that neutrophils from blood and neutrophils from the tissue can respond in different ways to the same pathogen. And this is particularly important for neutrophils because they're short-lived cells. It's not that they go into the tissue and they live there for months before being exposed to the pathogen, but they're very close to the blood neutrophils. So there are signals in the tissue that very rapidly regulate the neutrophils and modify their responses to viral infections. And my interpretation is that there's something in the genital tract that is preventing this neutrophil from producing calcium, probably because it is detrimental for reproduction, but that's to be demonstrated. So one question that is very important is, if we think that these neutrophils are protecting against HIV infection, we have to make sure they're located in the right place. Because if they're deep in the tissue and HIV infection is happening through the epithelium, by the time they get to the epithelium, it may be too late and infection is already established. So we started doing studies of the tissue, looking at the location of the neutrophils. And what we observe is that the neutrophils are located close to the epithelium, and we also have neutrophils scattered throughout the tissue. And we even identify some neutrophils that can go in between the epithelial cells and get into the lumen. So they definitely are located in the right place when it comes to HIV exposure. The question is, do all neutrophils respond the same to HIV? And when we digest our tissues to study HIV infection in vitro, we lose information of where these neutrophils come from. So we don't know if the neutrophils that are releasing nets are those that we see here or are those that we see here. And there's no way we can tell by digesting the tissue and then doing the in vitro infection. So we came with an alternative approach by using spatial transcriptomics. So when we do spatial transcriptomics, we stain for the neutrophils, which are here seen in red, In green, you can see the epithelial barrier. And then we can select areas of interest. So we select an area of interest of the neutrophils that are close to the epithelium compared to those that are farther away from the epithelium. And then we can extract the RNA from these neutrophils in each area of interest, and then do a whole transcriptome analysis. And then we compare the signal from those neutrophils close to the epithelium versus the neutrophils that are deep into the tissue. Are they different? So what we found is that they indeed are different. And there are a number of differentially expressed genes between both groups. These are ongoing analyses, but what I can share with you so far is that the neutrophils that are right here at the epithelial barrier have a signature for neutrophil activation, neutrophil degranulation, including different types of granules. They produce antimicrobial peptides, which are known to have broad spectrum antibacterial and antiviral activity. They produce type 1 interferon, also important for antiviral actions. And they have upregulated pathogen recognition signaling pathways. Again, the TLR signaling, toll-like receptor signaling that we've talked about. In comparison, when we looked at the neutrophils that are here, deeper in the tissue, we see a signal of differentiation instead of activation. We see the granulation, but only of some granules, meaning that they're secreting different products, the ones that are deep in the tissue and in the surface. We do see a signature for interferon-stimulated genes, and we see nothing for pathogen recognition. And that indicates to us that these neutrophils here, close to the epithelium, are probably receiving signals from maybe the microbiome and the bacteria that are present here in the surface. And they're acting as a first-line defense and first-line communication with the exterior. So in summary, I've presented that genital neutrophils can release nets that inactivate HIV and prevent HIV infection. That these are mediated through toll-like receptors. That there are different mechanisms in blood neutrophils than in genital neutrophils. That neutrophils are present in the genital tract in the absence of inflammation, and that we can differentiate those that are close to the epithelium versus those that are far away from the epithelium. And the two big take-home messages are that genital neutrophils and responses to HIV by genital neutrophils cannot simply be predicted from blood neutrophils. And probably for other infections, this is also true. So it's very important to study the cells in the tissue. And also we propose that nets represent a protective mechanism against HIV acquisition in the genital tract. And I'd like to finish with an updated model of HIV infection that takes into consideration the neutrophils. So now we know that neutrophils are there migrating into the lumen and also underneath the epithelium. As HIV comes in, it will find neutrophils that can release nets and inactivate the virus. Some virions may escape that, and then they have to overcome the epithelial barrier. They can be captured by dendritic cells, but they will also find neutrophils there that can release nets and other molecules to inactivate HIV. Some of them may escape, and then if they find a susceptible target cell, then infection will be established. But before that happens, there are multiple layers of protection that could explain why most women do not get infected after exposure. So with that, thank you to my amazing team. We were at Tufts University before. We moved here. Two of my students moved with me, but we are rebuilding the team. Also, thanks to collaborators at Dartmouth and Alabama and the funding source. And thank you very much for your attention.
And so in the older women, they don't produce the net as efficiently. Is that because of estrogen? And does it regulate TLR8 or?
So that's a great question. We actually looked, because that was my first hypothesis, is the estrogen, right? So for the blood data, we also had, we did estradiol detection in the serum from these women. and we correlated the net area with the estradiol levels, and there was no correlation. So we did see the decrease in estradiol in the postmenopausal women, and so the data looked good for the estradiol, but when we correlated with the net release, we didn't see a correlation. What I think is happening, I think estradiol is involved, but not directly. What I think is happening, I think as women age, there are senescent pathways that are reducing the functionality of the neutrophils. And I think those senescent pathways, the neutrophils are protected by estradiol from going into senescence. But as estradiol comes down, then senescence kicks in. That's my hypothesis, and that's where we're going with this, and we're trying to elucidate.
Thank you. Very nice talk. Actually, I used to study mother-to-child transmission of HIV in a previous life many years ago. And I was extremely interested in seeing your description of how low the risk per act really is for transmission. Turns out with babies is about the same. Babies are exposed every day, many times a day through breastfeeding. And in fact, you know, that was the premise on which I used to base my studies at the time. But my question is, do you know anything about the mechanisms you described in saliva, for example, or in the digestive tract that might be applicable to babies?
That's a great question. So actually, in the saliva is very rich in nets. So there is a very interesting study where they study neutrophils and net release in the oral cavity in healthy people. And they see that every day, there's a lot of neutrophils that come into the oral cavity and release nets, and they are part of our saliva, and they maintain health of the oral mucosa. And when you have problems with neutrophils and problems with net release, then you start getting ulcers and oral problems. So neutrophils play a key role in oral health. There's not much known about neutrophils in the intestinal tract, mostly because there's this dogma that neutrophils should not be there under non-inflammatory conditions. We know now that there are some neutrophils that are doing homeostatic functions, but that hasn't been studied in humans. It's all mouse work. So that's a whole field to be investigated.
Thank you. Thank you for your talk. So my question is, with respect to where this is going, I guess one of the next steps would be trying to see how to use this model for pre-exposure prophylaxis, is trying to prevent HIV, PrEP, as we call it. Currently, we only use tenofovir, is either TDF or TAF to prevent HIV PrEP. And the premise, the way it works is not this. So this is exciting. But looking at how this works, it looks like it takes up to 7 days to really get optimal activity.
No, that's I try to explain it. That's because of the in vitro assay.
Okay.
But actually, I incubate the nets with the virus, and then I add the cells, and then I remove the virus. So I am only measuring the virus that is able to get into the cell initially. And what happens is you need to follow those cells over time to be able to detect the viral growth. So that's why you measure at 3 days and at 7 days. At 3 days, you still have some of the background input virus. So if you don't have infection, you see like a flat curve, and you have some background viral proteins from your input. If you have viral infection, you see an exponential growth. So when we have the nets present, we see nothing growing. When we don't have nets, we see this exponential growth. And that's the maximum difference you see at 7 days or later. So that's why we have to do 7 days. But the virus is not there all the time. The virus is there for 1 hour. I incubate the nets with the virus for 1 hour. So within 1 hour, the nets inactivate the virus.
That was just my question, because if it's going to take that long to activate,
then it wouldn't be prevention. Yeah, absolutely.
Our next talk is from an external speaker, Dr. Jennifer Johnson. Dr. Jennifer Johnson is the first C.S. Mott endowed professor of public health at the Michigan State University. And Dr. Johnson is the Chief Translation Officer of the Michigan State University Office of Health Science. And Dr. Johnson is a licensed clinical psychologist who conducts policy-relevant implementation and effectiveness trials of mental health and substance use interventions for vulnerable populations, including perinatal patients. Dr. Johnson has been the principal investigator of a number of research studies worth $72 million funded by the National Institutes of Health, including 2 NIH Center grants. Dr. Johnson's research often uses community participatory designs and serves on local, state, and national boards and policy committees. It's our pleasure to have you here, Dr. Johnson. We look forward to hearing from you. Thank you.
While he's pulling that up, I am at the other end of the translational spectrum, but I'm excited to talk to you about it and why I do work there.
I was trying to see why it's not going into the presenter view like everyone else is. All right. Jay, do you know? Yeah.
Is everyone here a bench scientist? Is anyone not a bench scientist? Okay, all right.
You know, the idea is that the bench scientists also get a community study.
Right.
We need to talk different language, otherwise we don't know what we're doing.
Okay, perfect.
That's the idea of this meeting.
All right, awesome.
You educate us.
Okay,
maybe we educate you also.
Yeah, I'm learning things. Nets. I have a daughter who's a bench scientist. I'm gonna go talk to her about nets. Flex my knowledge. All right. Okay.
Do you mind if it's only on?
No, I don't care.
Screen? Okay.
And then, but it's just still forward like normal,
yes,
right?
So.
Okay, great.
I can duplicate it for you.
Thank you. All right. So I'm going to talk about implementation science, and I feel like this is not well understood necessarily even among clinical researchers, and I'm going to give examples from 2 different sets of studies. So here's our translational spectrum. I started my career kind of here as a clinical psychologist. The populations I work with, though, I work with people who've been involved in the criminal legal system and with pregnant women, typically lower income. So pretty soon I moved to real world clinical trials. But over the course of my career, I realized the systems in which I work are broken, and I could spend an entire career on interventions, but if the system has no providers or can't reach the people, it won't ever matter. So more and more I've been pulled toward these sort of larger scale, implementation policy research, because I really believe that if we had better health policy, we would solve half the health problems we have. All right, so what is implementation science? So it takes just 17 years for just 14% of knowledge and discoveries to reach common practice. So most of the work we do never gets out into the world. All this work, right? And members of disadvantaged groups are even less likely to receive evidence-based care or recent advances in care. So what implementation science does is it helps providers, systems, public health departments, whoever, do the things that we already know they're supposed to do, but we're humans and we don't always do the things we know we're supposed to do, right? And so this helps organizations do that. This I love, because implementation science language can be very meta and complicated, but this is the most basic description I've ever had, is whatever the innovation is the thing. We want them to do the thing. Effectiveness research looks at whether the thing works. Is it like hand washing or your drug or whatever it is? Implementation research looks at how best to help people or places do the thing. Okay. And then the strategies are the stuff we do to try to help them do the thing. And the outcomes are how much and how well they do the thing. Does that make sense? Okay. If you say an implementation science, if you say the thing, they'll know exactly what you're talking about. They will. Okay, so again, our outcomes, instead of satisfaction, function, symptoms, they're really acceptability, adoption. Do people use it? What's the cost? Is it feasible? Penetration is what % of your providers in an area use it, and sustainability is self-evident. Okay, so the research ingredients, there is a really good, this is like the Bible of how to write an implementation science NIH grant proposal. If you're ever interested, this is super useful. Last time I looked 2 weeks ago, there were more than 1,700 implementation science requests for funding at NIH. So if you're interested in this, it's super fundable. So the things you need, I'm going to walk through the steps using a couple studies as we go. So those are the main ingredients. Okay, so here's an example from an implementation trial that's finishing. So it's how much technical support is needed for agencies offering prenatal services to implement the rows. I'll tell you about it. It's 4 education sessions during pregnancy that prevent half of cases of postpartum depression. Right? There's not very many simple things that have that big of an effect. 4 classes during pregnancy, we can prevent half the cases. So, well, I'll tell you more about it. Okay, so here's the need. So postpartum depression affects 1 in 7 moms and 1 in 3 low-income moms. Chronic stress is one reason. Depression is very responsive to stress. And low-income mothers, it's especially concerning because they're not only more likely to get postpartum depression, but less likely to be connected to care. And if they get it, there's a lot worse outcomes for their kids. And yeah, so screening and treatment are important, but prevention is even better. And I'm sorry, I'll say one more thing about this. I watched, it came up on YouTube. It was a documentary about postpartum psychosis, which again can, we don't talk much more about the serious mental illness, but when you're sleep deprived, under a lot of stress, and it was something like 1 in 5 moms with postpartum psychosis die by suicide, and something like 1 in 5 try to kill their kids. And so it's, that's, for those of you who are out in the clinical world, really watch out for like postpartum mania and postpartum psychosis, because we don't talk much about them, and it's serious mental illness. Okay, so here's the thing. It's evidence-based, okay? So it's a 4-session class. It was developed by my colleague, Dr. Karen Zlotnick at Brown University. Her idea was, again, treatment's great, screening's great, but if we can prevent, wouldn't that be better? And she intentionally made it as a class to try to get around mental health stigma. And I'll talk more about that later, but it's a class, it's prevention. It was designed for prenatal clinics and other agencies offering prenatal services. In this implementation trial I'll tell you about in a minute, nationally we worked with 98 agencies, and they were everything from, we did have prenatal clinics, we did have, but we went as wide as like WIC, women, infants, and children. You guys know what that is, right? Or health education, or there's a great thing like a rise of like Black woman-owned doula services, like just pretty much anyone who is touching a pregnant person in some kind of healthcare or social determinants of health way. Okay, so the nice thing about our thing is, it has these core elements. And what I love about it, I wish I'd had this honestly when I was having my kids, because it's not exactly rocket science, but it's really powerful. It's the basic idea is, this is a time in your life you're going to need more help than you usually do. Don't try to be a hero. Just get that off. Just don't even go there. Let's talk about now what sort of support you might need after the baby's here. Get it in place now. And to the extent that the relationships in your life are problematic, sort of sort through that, who can be there and be supportive of you, and how do you make a sort of request? How do you talk to them now about the things you need? But the next thing, there's flexible things about it. You can see them group, individual, the order, it kind of doesn't matter. Doesn't require a mental health professional. We worked with a lot of visiting nurse programs, and if they're in there for 20 minutes, they can do it in little pieces, right? It's pretty flexible. All right, so care or quality gap. Prenatal clinics are scaling up postpartum screening and referral, which is good, but they don't currently do anything to prevent postpartum depression. We have our evidence-based practice, 5 randomized trials, prevents 50% of the cases. Our setting, like I said, we have 98 agencies across the country that enrolled in this trial. Okay, outcome, sustainment in this case. There's more, we need more implementation research in general, but we know more about how to get an agency or a practitioner to adopt something than how to sustain it. So we really intentionally focused on sustainment in this trial. So our question is, what's the minimum necessary outside support for agencies to sustain rows over 2.5 years? We know something about the facilitators and the barriers to use. It's a priority. When we did this sort of technical support with the agencies, what made me really sad is like a lot of these agencies that are serving low-income moms are they're like doing heroes work on a shoestring, but the guilt was just ubiquitous because they couldn't do more, right? So the facilitators are super motivated. The barrier is that they just are trying to operate on a shoestring. This is a conceptual model. It has to do with implementation science. Don't worry about that. It's an ingredients. We have one. That's kind of the point I want you to get out of that. Okay, so the study details. So there are 3 implementation conditions. And again, this is the stuff we do to try to get the people to do rows, right? So we had enhanced implementation as usual, which was basically 3 meetings up front. One, just sort of orienting the agency to what it was, what the logistics were, start to talk through who's going to offer rows. There was a clinical training. Well, we called it clinical, but it was really like sort of education for the community health workers or whoever it was that was doing rows. That was maybe 3 hours. And then I looped back with the office staff around billing or any logistical issues, right? So like it was a pretty solid, and we walked them through a plan, a sustainability plan, like who will do this piece, who will do that piece, who's going to bill, how is this going to work, right? And then we would give them a recording of all of those meetings so that if they had turnover, they could train someone else. So it was about the best you could do at sort of a, in just a few weeks. And then our other implementation conditions were quarterly coaching and feedback and monthly coaching and feedback. And as you can see, our outcomes were how long was rows sustained with adequate fidelity, reach, number of people getting it, health impact. And again, this is, we already know this is an evidence-based practice. So this is not really where we're doing our level of control, right? It's not sort of randomized on that, but it's nevertheless good to sort of measure it. Cost effectiveness, and agency capacity and ownership, because there really isn't much known about what's the return on investment of this sort of technical assistance. So yeah, this is the most complicated study design I've ever had. In implementation science, you can use a randomized design, you can use any of a number of designs. This just happened to be a sequential multiple assignment randomized trial, which was, we started with, this says 90, but it was 98 agencies. Looked at 3 months, every 3 months, were they either not offering rows or not offering it with adequate fidelity? So if they were sort of failing to sustain, that was the risk. So if no, they continued in this regular, this EIAU, which is just no further intervention. If yes, they could be randomized to either continue or to get this, quarterly coaching and feedback. And again, if at a subsequent quarter they were at risk, they could be randomized to either keep getting quarterly help or get monthly help. It's basically just sort of a dosing thing. Okay, so I will tell you, we haven't done the final analyses yet, but it was really interesting because the results are very different than our felt experience offering the technical assistance. About 40% of the agencies actually could sustain with just that first, which was more than we thought. That's actually really good news. And then we sort of thought it was that quarterly piece that helped, but so far there's a difference for the agencies that were struggling. It looked like the monthly was better than the quarterly, and the quarterly may not have been better than just the original. But one of the cool things you can do in this kind of analysis that our statistician is still working on is figuring out what kinds of agencies need what level of technical support, right? So she's gonna do that. We don't have that yet. But what we found out was, we didn't have really randomizations for low fidelity. What was really sad, like I said, it just sort of broke my heart, is even agencies that were maybe hospital-based, which were better resourced than like a volunteer doula group or a lot of other things, their problems weren't with rows, their problems were with keeping their doors open. And it would be something like the hospital would say, oh, your Medicaid clinic for moms, oh, sorry, didn't we tell you you need to move space? Oh, it's a quarter of the space. Sorry about that. I guess you'll have to close down some of your programs, right? It's just, even the well-off clinics, it's just really hard to do this work. And I don't honestly know if, we say that we care, but I'm just telling you, structurally working with 98 agencies across the country, they're not supported like we care about these ones. Okay, so, support the agencies. There's a whole thing around the billing codes that I could tell you about. Basically, under the Affordable Care Act, this is a U.S. Preventive Services Task Force Level B recommendation. which means it's supposed to be supported under the Affordable Care Act by law. The problem is in each of the states, there's only one state where I think it's really easy to bill for this, because the problem is that there's several problems. One, that people who can do it, like nurses or health educators, are billable providers. Two, if it's a mental health professional, they can't bill under a pregnancy diagnosis code, which is how a physician bills pregnancy preventive care. But like as a psychologist, I have to give you a mental health diagnosis, and the point of prevention is you don't have a mental health diagnosis, right? So that doesn't work. And then we have these education codes which aren't reimbursed by Medicaid in a lot of states. So we've been working on trying to fix that, because by law it's supposed to be reimbursed, but pragmatically it's really hard. And we found that it's better if it can be integrated into the regular workflow. We actually have a second R01 we're just studying, because what was really interesting is NIMH put out a request for abstracts that said, what we found is we can prevent postpartum depression among high-risk moms. Should figure out who's high risk. But we knew from doing this trial that actually most of our agencies offered ROSE to everyone. And the reason for that is, one, it's just easier for them, even the ones in the hospitals, to just give it to everyone because it's 4 sessions, than it was to try to do screening, referral, then you got to follow up with people, right? It's a whole thing. Secondly, our most marginalized moms don't, like we had a clinic in Texas, and our undocumented moms really didn't want to be told, oh, we think you're at risk for depression or something, we're going to put you on a list. They do not want to hear that. They think they're identified, this could be reported to the government. So the more marginalized the group was, the less likely they liked being singled out. And our health economist did a simulation study and found that there would have to be essentially 0 false negatives for screening to be cost effective anyway. Because the cost of an untreated case of postpartum depression is about $30,000, even if you just look at the health system costs, and the cost of the intervention is about $150. So we responded to this and said, with saying all of that, and said, oh, we don't think that's the right answer. And so right now we just started it's a 2,300 woman randomized trial looking at, the effectiveness, the cost, the equity of offering it as universal prevention. Okay, so I will give you another quick example. I'm going to talk about maternal morbidity and mortality disparities. So in the US in general, maternal mortality is higher than other high-income countries, and it's going up while others are going down. More than 80% of the deaths are preventable. What I think people don't realize is how many occur during pregnancy, way before the hospital or in the year afterward. A lot, some are happening in the hospital, but it's a lot after. There are disparities, and what's interesting about the disparities is when you control for site of care, pre-existing conditions, health behaviors, there's still a lot of disparities. So there's things going on that you can't blame the mom for or other things for. How many of you have read this book? Okay, this is literally my favorite book. It was given to me by my partners when I came to Flint. And it's an anthology. It's just like basically a qualitative study, but it reads like a novel. And it helped me really understand a lot of the cities in which we're working, because the gist is from the 1920s to 1960s or so, a lot of folks fled the Jim Crow South because they were going to get killed because they were saying things, right? So they came to the North, and in the North, it wasn't quite like they were going to get killed, but it was like a lot more, it was not great either. So what happened in Harlem and South Chicago is, they said, okay, you guys can live in this 10-square block area. By the way, there's 200,000 of you. Sorry about that. We're going to triple the rent because you can't live anywhere else. You're not going to get jobs, right? So it was just so hard for people to live in the North that, while the parents were just trying to survive, in a lot of cases, the city got the kids, or we created, like South Chicago, we created all of these neighborhoods that we talk about now, like, oh, but like really racist people in the North did that. So it's super interesting, highly recommend. So, some of the effects of maternal mortality disparities, I'll go over them super quickly. At the community level, for a lot of reasons, African-American women are disproportionately likely to have poverty, live in neighborhoods with problems about built environment. In Flint, we have a lot of neighborhoods that don't have streets, but also the emergency manager sold the snow plows. So, you have a mom with a stroller and no sidewalks and no plowed streets trying to get to a point. That's really hard. Safety of buildings, having to work multiple jobs, etc. System level, Hospitals serving more African-American women tend to offer lower quality care. And it's interesting, California did this whole hospital-based quality improvement initiative, and they improved quality of care overall and didn't affect disparities. Provider and practice level, sort of, and I don't think most or any providers get up in the morning thinking, I'm going to be racist today. But the truth is, you know, we act a little different depending on who's in front of us. We do. It's a human thing to do, and they don't get the same care. And when even the front desk staff is rude to you, do you go back? No, you don't. So then people are like, well, why aren't African-American women coming in for prenatal care? And it's, you know, because they experience really negative things in the healthcare environment. Have any of you seen around Serena Williams, her birth story? Okay, well, so
pregnancy complications.
yeah, she did, but it's the same, yeah, but it's the same story that they tell of like people in the hospital. Like it just gives you goosebumps. If people in the hospital, like there was an African-American woman, she was bleeding into her IV. Her husband tried to get the nurse to pay any attention. They just sort of blew her off, and like 10 hours later, she almost died. Yeah. Right, it's the same thing. And so being rich doesn't protect you. Being you know, famous doesn't protect you. If you're a Black woman in America, you are not going to get hurt in the same way, and that's scary. So unintentional biases, research has shown that controlling for patient class, health behaviors, comorbidities, insurance, healthcare access, healthcare providers undertreat African-American patients for pain. This is in general, not just in obstetrics. They listen to African-American women about their symptoms. I love this quote. There's so much judgment. If you're too calm, then they say, oh, you're not sick. You don't look sick. And if you're crying and moaning, they say you're exaggerating. I really don't know what they want from us. And like I said, there's just story after story about mom saying something is really, really, really wrong and just getting blown off by the healthcare system, and then mom dies or something bad happens. They blame them for their symptoms, provide less appropriate care. I have a friend that does work in endometriosis, and you know, if an African-American woman comes in and complains of pelvic pain, she's more likely to say, oh, it's a sexually transmitted infection than it's, you know, some other thing. Older, cheaper, more conservative treatments discharge African-American patients too soon and prescribe inappropriate medication. And I'll say, you know, a lot of people sort of see the root of all of these causes is sort of just underlying structural racism and unintended racism, but the truth is people don't get treated the same. So what can we do? I have 7 minutes, I will tell you. So here's 3 questions. How can you help services be most responsive to moms' needs? And how can you better engage those moms in improving the healthcare systems that serve them? They know what's wrong with your system. You know, go ask them. You know, have them, you know, as your partners in fixing this. And I have to say these slides, I'm going very fast through. It's an R01 and now a center, but I wrote it. My thing isn't really maternal health equity, but I wrote it because our partners in Flint had been telling me since I came here, they really care about this work around infant health disparities, and they knew what to do about it. So when we saw the call and I knew they wanted to do it, I was like, all right, I know how to write grants, let's do it together. So a lot of this I'm pulling from, you know, things that they wrote or presentations or whatnot. So, you know, involve them in improving the care. And if you can't do everything, do one thing. So my point here, you know, I could send any of these to you. There are some good articles having recommendations, you know, in communication. I like this disparities dashboard. The idea is if you're in a system that has any sort of numbers capability, not all systems do, look at your outcomes, whatever, to pick something, birth or preterm birth, or I don't care what it is, pick something. Look at the disparity, and then if there's a disparity, make it your own fault until it's fixed, right? It's not like, oh, they don't do this, or oh, they don't, no. It's your fault until it's fixed. And so that's sort of the dashboard idea, right? Equity, and again, you know, the moms know what's wrong with, you know, I just ask them. And it's not always the doc, like I said, nurses, front desk staff. One of the things we're trying to do in this is train everyone in the health systems, you know, that interacts with mom. Here's another model, you know, really looks at interconception health. My point here is that there's lots of things you can do. It's not hopeless. I really like this blackmamasmatter.org toolkit. If you're interested in policy, they have a lot of good tools around policy advocacy. Like, for example, expanding Medicaid. I think Medicaid covers something like 43% of Michigan births, but it's more than 60% or maybe even higher births to Black moms, might even be higher. So again, it's sort of this right to health, physical accessibility, economic accessibility. Again, take home point is there's things you can do. So we have an R01 right now that's in its 3rd or 4th year, where what we're doing is looking at a few different levels of things, but one is training, sort of actionable maternal health equity focused training for everybody from the front desk staff up to the providers and everything else. But it's really focused. And the other thing we're doing is there are some quality improvement bundles by the Alliance for Innovation in Maternal Health, Community something initiative, and Community Care. And so these are different than a lot of the quality improvement initiatives in that they focus mainly not on the hospital, like in this before or after where 80% of moms die. And they also focus on equity specifically. So it's not just overall quality, it's things you can do to improve equity, some of the things we just showed. So that R01 is ongoing, and it's a quasi-experimental difference in difference design, which I could tell you more about, but it's well-controlled of 2 counties, Kent and Genesee versus the rest of the state. We just got funding from implementing this big NIH. There's 10 maternal health centers of excellence. And the gist is, in this center, we have 3 different projects that we're testing. And it was really cool. A colleague, Steve Undersma, had this idea of, we layered the projects in a way, these are all different, you see in the counties where there's some in their own counties, but all the projects overlap at least one county so that we can look at the independent and additive effects of all of them. So what we can do, and we're measuring cost data, we're measuring everything through Medicaid claims. So we're intervening like sort of with all the different systems in the county, but we're using population level claims data to look at effects. And by overlapping the counties like that in this partial factorial design, we can look at the independent and additive effects of the different interventions and then say, if you want this, it costs this much kind of thing. And this is a horrible acronym we made up, but we didn't know about. So basically, pregnancy associated means it can have to do with not direct physical results of pregnancy like suicide, overdose, homicide. Black moms are actually, even though they get drug tested more, way less likely to die by overdose during pregnancy and postpartum, equally or slightly less by suicide, but much higher by homicide. It's very risky to be, a woman in general is most likely to be killed by her partner pregnant or postpartum, but for Black women, it's like 8 times as much. So anyway, so we're looking at all of those deaths, all during pregnancy and postpartum. And then the part, that I wanted to tell you about with this implementation science is, so this is another implementation science design, and it doesn't have to be this big. It could be like, I have 200 providers, right? But like I said, for me, more and more over my career, I'm convinced that these structural issues are the problem. So the studies I've been doing have been getting sort of at this bigger and bigger level. So in this study, basically we have these equity-focused quality improvement things. It's the thing. We have 12 counties, stepwise design. How many people know what a stepwise design is? Do bench scientists do that? Okay, all right, I'll explain what that is. So it's where you have, everybody gets the control in their intervention. They do it in this case in 4 steps. There's 12 counties, but you randomize them to the steps, and then you measure everyone continuously. And so it is a randomized controlled design. It's just, it has better power when you have like 12 counties versus randomizing 6 and 6. And with nearly 6 million people, and we're gonna test, see if we can do these. It's sort of basically technical assistance again. If we work with them and, we'll characterize the technical assistance we do give them. Can they adopt these equity-focused quality improvement initiatives, and do we see effects on the outcome, and what does it cost, and what's the benefit, right? So that's the sort of large-scale question. Like I said, this is a little risky because I'm used to like a more hands-on randomized trial to sort of intervene out in the world and then hope you see population-level effects. It's a little risky, but it's important, and so I hope it works. Yeah, and that's my talk. So again, what I want you to get out of this, 2 things. We can prevent postpartum depression. We can address maternal mortality and morbidity disparities for those of you who are clinicians, or just people, think about some of the ways to do that. But third, to just sort of understand this new kind of methods around implementation science, because I think it's powerful, and I think, well, like I said, we can create the things that help people all day long, but if they never get to the people, it doesn't really matter. So that's why I do what I do. Thank you. So I finished in time for questions. Yeah.
Very good. No, thank you. Biologically, there is something that is called the memory. We transfer into our children epigenetic modifications, and it becomes just cycles. Preterm birth, for example, it's a cycle that passes through generation after generation. biologically, we cannot stop that, unfortunately, at the same time. And that is the reason I also leave it to you, how we can intervene in order to stop these cycles that are repeating year, generation after generation.
Yeah, and so I mean, so it is, you're right. The epigenetics are there, but what we can do is with the mom we have, try to reduce the stress in her life as much as possible.
Because that stress is passing to the daughters.
Right. And when I asked our partners the things that they would do if they were in charge of the world, they said, universal minimum income, good, time off and all these things, but anything we can do to support mom. Like, so, again, I hate going back to this documentary on psychosis, but it just illustrated so many things. This mom was floridly psychotic, killed 3 children, 2, 1, and 0, but she had not slept in weeks. Her husband had this sort of idea that his job was to make money, and so she should deal with all the kids. She had a trauma history. She had, like every kind of stress, and nobody was there. And now her kids are dead, and she's serving a prison term because, there are these heartbreaking calls of her trying to call her mom, saying, "Mom, something's wrong. I don't know what's wrong. Everything's weird. I can't sleep. I really need help." Right? And then within 24 hours, the 3 kids were dead. So I mean, I hate to use that graphic example, but we just leave pregnant moms and postpartum moms in such a lurch. Like anything you can do to reduce stress. So whether that's as a physician, whether it's around social determinants of health, whether it's talking to, I think part of the reason, the reason Rose works is we talk to the people, help her talk to the people in her world about getting support in place, anything you can do to help them sleep. Like, I again, I'm a psychologist, they come from psychosocial, but I'll tell you, I used to work in a partial hospital program for mental health disorders in pregnancy and postpartum. And I would see moms that would literally wanted to die on Monday. The psychiatrists who work in pregnancy and postpartum are way more likely willing to prescribe to moms, mental health medications, and you would give mom sleeping medication. And by Friday, she was almost fine. Right. So it's not just, it's like, we really, I don't know, anything you can do to help reduce mom stress, whatever that looks like. Yeah.
So I was wondering, is there any correlation between the length of maternity leave versus those terrible impacts of mental health postpartum?
You mean how many weeks?
Yeah. Because in the US it can be nothing versus 12.
Oh, you mean maternity
leave postpartum?
Yeah. I mean, I think
countries 1.5 years.
I think there is some evidence around that. I think it's mixed and not super strong. I will say this, I'm really excited. So I really wanted to put this sort of universal income thing in here, but we had, these centers, they give you 10 weeks and I couldn't come up with the money and I couldn't figure out how to do it. So I wrote it into this project and then I had to write it back out again because I couldn't tell NIH how I was gonna do it. I didn't know. In the meantime, turns out Mona, who also is in Flint, had the same idea, but she didn't have to deal with NIH, and she has raised more than $40 million to provide a universal income to pregnant and postpartum moms in Flint. And so because we had the evaluation all put together, she was really thinking about the kids, but she sort of had that idea and I had that idea, but I couldn't figure out how to explain it to NIH. So she found the money to do it. So we can also evaluate that sort of income piece around it, just the financial stuff. But sleep matters, income matters. I can't imagine having a 2-year-old, a 1-year-old and a baby and a partner that didn't see it as their job to help me with that. Yeah, go ahead.
I saw that your next goal for the ROSE program is to try to get involved with U.S. preventative task force. Maybe could you collect any secondary, tertiary data explaining breastfeeding rates improving or improved infant outcomes? I think it's 2.5 years out. So do you have baby data as well? We
do not have baby data, but I'll tell you what's going to change U.S. So U.S. Preventative Services Task Force recommendation is what it is because all the randomized trials of ROSE, and there's one other, were done in at-risk samples because then you only need 300 moms, you don't need 1,000 moms, right? So it was purely an artifact of being able to do the trials, but you just need a higher base rate to power the trial. So basically what we said is, we have no evidence if this works or not, but we have all these reasons to believe it's more pragmatic in the real world to give this to everyone, might be more cost effective and more equitable. And so in this 2,300 mom randomized trial, we'll be able, it's universal, it's all takers, and then we randomize everyone, but we also have a bunch of potential things that people might have in the medical record that could indicate risk, like Medicaid status and past depression and some other things. And then we can look at, whether or not if you cut the sample by any of those things, Is it equitable? Is it not equitable? But the reason U.S. Preventive Task Force has said what they said is they don't have evidence to the contrary, and the reason they don't is it's just a big trial. So we said that to NIMH, and they gave us the money to do a really big trial. So that's the, and then I think if we go back to them with that, they're totally on board. Yeah.
Just with early career clinicians and scientists here, how would you say was your process in getting to implementation science?
So like I said, you know, implementation science, it's not really new. It was going on in the 50s with farming. It's newer to healthcare. I learned about it during my K award. So my K award was funded in 06. So 17-ish years ago, I learned about it in 08. It's pretty new and growing field. And so what I would say for an early investigator is I do a lot of things now that I was not originally qualified to do. But the thing is, you get a collaborator on a grant. For example, I have my favorite health economist, right? Or an implementation science co-I. You do the grant, and then suddenly, like, the case you have to make when you write the grant is the team has the experience. But then once you have the grant, you can sort of say, I've done work in X or Y. So I would say get a good implementation science co-I or two. There's also online trainings. I look up TIDIRC online trainings. There's some good books. If you email me, I can give you like my top 10 articles, but learn about it. And don't feel like you're the only one who doesn't know, because truly it's newish field, like past 20 years, it's evolving all the time. So nobody really, I mean, a few people really know, but you know, you can absolutely hop on this train, get people who know how to do it a couple times, and then you know how to do it. Anything else? All right, thank you so much.
It was an amazing presentation we had. We're very fortunate to have that seminar here at the CS Mott Center. So we're going to take a short break, and we will meet here back at 3:30. And following which, we're going to have a famous epidemiologist that's going to talk to us. And thank you very much. Okay, 3:30, we'll be here. So let's get started for this the last part of this session for the symposium. And before we get started, I also wanted to say that at 6 p.m. we have something called as the Talk with the Meet the Experts, and this is a Zoom session, except for the speakers who are going to be here in this room. And this is a session where we're going to be interacting with public. It's been advertised, and we're going to take questions through Zoom from the public. It's going to be live streamed and it's podcasted as well. And I would appreciate if you could join for this discussion, and we'll try to send the Zoom link once more and the password to to log into the session. So this we have only 2 more talks. Very excited to introduce to you our next speaker, external speaker, Dr. Eduardo Villamor, who received Dr. Villamor is a professor of epidemiology at the University of Michigan, Ann Arbor. And Dr. Villamor received his medical degree from the National University of Columbia and a Master of Public Health from the Hebrew University of Jerusalem, Israel, and a doctorate in nutrition and epidemiology from the Harvard University. Dr. Villamor is an assistant professor for the international nutrition at he was an assistant professor at the international nutrition at the Harvard School of Public Health from 2005 through 2009, after which he joined the University of Michigan at Ann Arbor. Dr. Villamor has lived and conducted research in the Middle East, in Africa, in Latin America, and currently serves as the director of the summer session in epidemiology. Thank you so much. Really appreciate having you here.
Thank you so much for that. I want to pay your kindness forward for inviting me with this cartoon from the New Yorker, which used to be our self-deprecatory introductory card before the pandemic, after which some epidemiologists became famous and kind of being an epidemiologist became a little bit more fashionable than before. Thank you for inviting me. Well, 3 weeks ago, a baby was born in Canada. Possibly many babies were born in Canada, but this one made it to the news. And that caught my eye, and even though it's false, it caught my eye because the reason why the baby made it to the news was because he broke a record on birth weight. The baby was born at, if you are into the SI system, 6,600 grams, which is about twice as much as a normal weight baby should be. And the one particular thing that was a bit shocking, I have to admit, was the celebratory tone of this event, because there were bets and there was cheering when the weight scale hit that number. So, being a perinatal epidemiologist, well, this is an adverse pregnancy outcome. Obviously, you don't cheer for adverse outcomes. So the doctor who delivered this baby was interviewed, and I found it very interesting. Also, the way this doctor understood the situation. So when he was asked to say what makes babies big, he said, well, in general, what predicts a big baby is genetics, post-dates, and poorly controlled diabetes. That's absolutely true. But to be sure, this particular mom did not have diabetes. And then he just concluded, the mom had another big baby before, so he just said, I think she simply makes big babies. This is also a reasonable conclusion because, as we know in epidemiology, individual causal effects cannot be determined, so that's okay. But I think he still sort of missed the main problem here, which is this: the most important risk factor for macrosomia is maternal obesity, even irrespective of whether the mom develops diabetes or not. And the problem is so dramatic, really, that it can become a vicious cycle in that basically a mother with obesity has high risk of having a big baby, and that big baby is likely to become, if a girl, a mother with obesity herself. So perpetuating that vicious cycle of obesity and risk. Now, the problem from a public health perspective, I guess we all know, is a serious one. I guess for its dramatic effect, if any, I brought here a very simple graph from a study we did almost 20 years ago, actually, when I was living in sub-Saharan Africa, and documented for the first time then the advent of the obesity epidemic among pregnant women in a region that had been, I guess, typically associated with the other side of the problem, with underweight, undernutrition, etc. But at the time, we noticed that coinciding with the turn of the century and the new millennium, there was an increase in the prevalence of obesity in early pregnancy among women of threefold, from about 3% in the mid-1990s to almost 10% 10 years after. Here in the US, the last CDC survey tells us that at least 31% of women arrive at pregnancy with a BMI of 30 or more. And obviously, that is an average number because there are huge disparities in that number as well. And in some subgroups, that prevalence is much higher. So, going back to the big baby case, macrosomia is not the only and certainly not the worst possible consequence of maternal obesity. And I brought here a summary of the very first epidemiologic study, really, the clinical epidemiologic study that I could find back from 1938, and in fact it's in German. You know, at the time, German was the scientific lingua franca. So I have translated some of the words in German to English here, because of you who may not speak German, like me, like I do not speak German. But to describe the study, basically what this doctor did in at the Riks Hospital in Copenhagen was to compare women with overweight. It was 242 women with overweight with a group of 484 women without overweight. And the definition of overweight was according to prevalent standards at the time, had to be about 20% of the mean. The maximum weight of normal weight woman was 75 kilograms, and the minimum weight of overweight women was 84 kilograms. He did try to separate the groups. And basically what he discovered was that the birth prevalence of pretty much any outcome could look at was much higher in women with overweight compared with the group of women without overweight. So that's basically what you can see here. So prolonged labor, uterine inertia, I guess preterm labor, assisted delivery, cervix cut, clitotomy, which is you know, a practice that I guess from the history of obstetrics, our colleagues could probably tell, but we probably don't want to remember that. Basically traumatic delivery and hemorrhage, macrosomia, and stillbirth. Now, many studies have been conducted after that. I have to say pretty similar in the basic design, obviously with fancier methods, larger populations, etc. And the conclusion has been quite consistently the same. Now, as epidemiologists, as lame as that may sound and be, we are actually always concerned with whether any of these differences have a causal interpretation. That's what we care about, because a lot depends on it. So most studies resembling this design, you know, will really perhaps improve a little bit the possibility of making causal inference, but only incrementally. The only way to find out whether maternal obesity causes these outcomes would be to conduct a randomized trial in which you assign some group of women to obesity and another group to non-obesity. But that study is not happening anytime soon, because to begin with and end with, no IRB will ever approve it. And obviously, I guess no investigator in their right mind will attempt to do that. So we are left with tricks that we can attempt to implement as epidemiologists in observational studies, meaning we don't try to intervene necessarily, but can use those tricks that, you know, the methodologists studying causal inference have provided us with. So we used one of those tricks. We took a look at this problem from a slightly different perspective, using population-based registries from Sweden, which I will describe in slightly more detail in a few slides. But basically, these registers include the whole population with the characteristics that you decide to restrict them to, and are very large and are very high quality. So the difference in the approach that we took to the study of maternal obesity and pregnancy outcomes was to examine what would be the relation of weight change between pregnancies and the risk of having an adverse pregnancy outcome in the second pregnancy. Studying change in an exposure, it's usually stronger and closer to causal inference than studying the exposure at a single time point, because change takes us closer to what we currently in causal inference consider the gold standard of causation, which is the counterfactual, meaning what if a woman, instead of having this weight, had this weight. It's not perfect, obviously, as we'll see, but it takes a little bit closer to causal inference. So on these graphs, I have on the x-axis the different categories of the amount of change in BMI that women experienced from the beginning of the first pregnancy to the beginning of the second pregnancy. So, for example, less than -1 means women lost weight from one pregnancy to the next, and then you have the consecutively increasing categories. And then on the y-axis, we have measures of risk. In this case, is the relative risk. So each square here, each box represents the risk, the proportion of people with the outcome over everybody at risk in that category compared to the same number in the reference category, which is always women who maintain more or less a constant weight between pregnancies. So here is the case of the big baby that we started with, large gestational age. You can see those response association between the amount of weight women change between pregnancies and the risk of having a big baby in the second pregnancy. Of note, if the mom actually loses weight between pregnancies, the risk will be lower than if she remains at the constant weight. Then you have a similar pattern for cesarean delivery. By the way, the big Canadian baby was delivered by a C-section, and you know, these two events go hand by hand because it is very hard to deliver such a big baby vaginally. There is also an increased risk in gestational diabetes with weight gain. There is increased risk in preeclampsia, and there is also increased risk of preterm birth, both medically induced, typically through preeclampsia and to some extent gestational diabetes, but also spontaneous preterm birth. So we also extended this analysis to the perhaps the ultimate and most serious outcomes of pregnancy, at least from the offspring point of view, which are survival. And also weight gain between pregnancies is related to increased risk of stillbirth. And if we extend even further the analysis of survival among live births, it is also related. There is a slight signal here for increased risk of infant mortality, defined as death within the first year of life. There have been other studies that confirm these findings and again support the notion that there may be a causal link between maternal obesity and the risk of adverse pregnancy outcomes. Now, I'm going to present what we have been doing since using causal diagrams, and sometimes this is hard to sometimes I try to sort of explain the basics here. The rules are actually pretty basic, but the arrows represent causal effect between variables, which are the other components of the diagrams, right? So in this case, I'm calling F1 BMI the BMI of a mom, and I'm calling F2 SB the adverse outcome of the offspring, and SB in this specific case is stillbirth, because this is, you know, being the most serious outcome is what we concentrated on next. So in this particular causal diagram, what's encoded is a causal effect of maternal BMI on the risk of stillbirth of that mom's baby. Okay. Now, BMI has a strong genetic component, and it is reasonable for us to assume that eventually some of the genes involved in BMI might also be somehow related to the risk of stillbirth. Therefore, I added a new note here, which I'm calling shared factors, including probably mostly genetics that might be a common cause of both the mom's BMI and the offspring outcome. When you have a common cause, then the initial arrow of interest no longer has a pure causal interpretation, because there is some association that's not causal, that's going through this path, which we call backdoor path, right? So in classical epidemiology, and still it is called confounding, right? Okay. On the other hand, obesity also is strongly heritable. Therefore, one could wonder whether there could be an association due to the inheritance of BMI with the BMI of the previous generation, meaning the ancestors of this mom. So this path now linking, for example, who would be the grandmother of this offspring, being associated statistically with the outcome of the offspring, even though this path does not go for a causal association, because for an association to be causal on this diagram, all the arrows have to flow head to tail from the distal variable to the outcome. But that doesn't rule out that there actually could be a causal association between the distal ancestors' BMI and the offspring pregnancy outcomes through possibly the mom's BMI, or maybe even more interestingly, that there could be a direct effect somehow of that distal ancestor on this grand offspring outcome through exposure of the mom perhaps to obesity of the grandmother in utero. That's what we set out to study. Basically, these two paths, which would be the potentially causal paths, that might link maternal, grandmaternal BMI with the distal pregnancy outcomes of the grand offspring. And we did this using the Swedish nationwide registers. The backbone of those registers is the Swedish National Birth Register that was initiated in 1973 and has pre- and neonatal information for 99% of births occurring in Sweden. And it's standardized, and by law, every single hospital and maternity has to send a copy of this information to a centralized register. And therefore, it can be a paradise for perinatal epidemiologic research. Also because you can link the information on that register with that of other registers, such as the patient register, which was started in 1987 for hospitalizations and 2001 for outpatient visits. So every single doctor visit that occurs in Sweden goes to a centralized register. You have a cause of death register, you have a population register with dates of immigration, you have a multi-generation register that tells you who is first relative to whom, an education register that has the educational milestones of everyone there, and everything can be linked to a national registration number that's assigned to every person in Sweden at birth or at immigration. So again, it's really a unique and wonderful resource to work with in epidemiology. So using information from these registers, we assembled what we call the 3-generation cohort. that comprises a maternal grandmother, the F0 generation, a mother, the F1, and the grandoffspring. And here I have some basic characteristics of the cohort. This is the sample size for each generation. So obviously, there is more grandoffspring because some moms have multiple kids and some grandmoms have multiple moms and daughters and grandkids. These are the birth years of each generation. And here I have the mean BMI in early pregnancy, which is measured at the first prenatal visit, which happens in Sweden on average at week 10 of pregnancy. And you can see the difference in mean BMI between the two generations, right? So the population level, look, this is a huge increase, for example, the prevalence of obesity. I have to say it's not as explosive as it is yet, as it is already in the US, but still you can see a fourfold increase in the prevalence of obesity. Okay, so back to our research question of trying to measure really the size of these two causal errors. Well, we have a problem because due to this sort of lurking variable here, we also have non-causal paths. We have at least two non-causal paths between this variable and this. I have highlighted here in red one of those non-causal paths, and we have another non-causal path here. Therefore, we have to do something about that. The problem is we don't have data on this to adjust for, to control for. So we have to start thinking of those tricks that have been sort of proposed lately by the causal inference investigators. One of them, which could be implemented in this case, is what we call negative exposure control. So negative exposure control is a variable that cannot be possibly have a causal effect on your outcome, but that can be and should be associated with the outcome statistically through non-causal paths that are shared with the actual exposure of interest. And we found a really good negative exposure control in this scenario, which could be the BMI of this mom's sister. So the BMI of a sister, of a mom's sister, should not have any causal effect whatsoever on the sister's risk of stillbirth. Yet it is statistically related to that through non-causal paths that are also the same causal paths through which this distal exposure is related to your outcome of interest. So basically the idea is that after you do your analysis and you control for what you can control, you should not see an association between your negative exposure control and the outcome. And if you still see an association between your actual variable of interest and the outcome, then you might have successfully closed the backdoor non-causal paths, and maybe that would enhance the chances that the association you are seeing is actually causal. So that's what we did. Now, the causal diagram is a bit more complex, and just to basically scare you a bit, it's splendor, but again, just to show that we had to adjust for many more variables and do sort of more analysis and stuff. But it's kind of complex. Okay, so I'm going to show the results now on the associations between maternal grandmaternal BMI in early pregnancy and the grand offspring risk of stillbirth. So I'm going to be using similar graphs as before. And here we have that same association I just announced. So on the x-axis you have BMI, and on the y-axis you have risk of stillbirth. This dotted line at 1 means the value of no association. So everything that's above that line means there is increased risk. Anything that's under that line, there is decreased risk. And the reference value is the BMI, the median BMI for the population, which was about 21.7 at the time. So basically what this particular panel shows is that compared to the median of the population, there is increased risk of a stillbirth in grandoffspring. And there is a dose response association again above the median value. These dotted lines, like the whiskers, are the confidence bounds of this association, meaning when if both of them are above or under the null line, that means the association is statistically significant, right? Okay, so basically what we have here is that there is a positive association between grandmaternal BMI and the grandoffspring risk of stillbirth. So for comparison, I brought here the association, the same population with maternal BMI. I mean, this we already knew, but again, to contrast the size of the association. So they are relatively comparable, perhaps slightly less for grandmaternal BMI, but still pretty consistent. And then finally, I brought the association with the sisters, the mom sisters BMI, which again is the negative exposure control, and we should expect no association if we have closed the backdoor paths that are non-causal properly, and that's actually the case here. There is really no association going on whatsoever. So that means there might be an actual causal association between grandmaternal BMI and the risk of grandoffspring stillbirth. The next part of the analysis was we tried to decompose that overall association into the two causal pathways that I showed were of interest at the beginning. This one, which is called an indirect causal path, and this one, which is called a direct causal path. And we do that using some new methods that are called the causal mediation analysis. And when you do that, what you find is that in this case, less than 1/5 of the overall association is mediated through the mother's F1 generation BMI, which means that the majority of that association is either direct or indirect through paths that we did not consider. But this is interesting because one of the possible mechanisms, again, why this might be happening is exposure of the mom to the grandmom's, her mom's obesity when she is in utero. So, you know, of course, I am no epigeneticist by any stretch of the imagination, but one could imagine that being in a intrauterine environment, you know, where there is inflammation, etc., because of the obesity might possibly affect the germinal cells of these fetus. I don't know. And then we are working on these. These are still unpublished results, but I also looked at infant mortality of the F2 generation in relation to BMI of the F0 generation. And as you can see, there is also a strong positive association here for grandmaternal BMI. There is also an association with maternal BMI, although I guess this is somewhat stronger. And using the negative control again, there is no association with the sisters' BMI, which again indicates that there might be some causal interpretation to this association. And when you decompose the effect for infant mortality, as opposed to stillbirth, most of it, at least 2/3, is through maternal BMI, and only 38% is talking. So those are the results I wanted to share, and here are some thoughts on this. So one is the implications, and this is a difficult reflection because obesity is a very heterogeneous condition of very difficult treatment and even more difficult prevention. But you know, there are some interventions, for example, to prevent weight retention postpartum, and they are relatively effective, although the size of the effect is usually modest, but they exist. Earlier prevention of obesity obviously would be ideal, but again, that is a very challenging thing to do for clinicians and public health practitioners. But the other is, sort of thinking of future, what are the mechanisms? So as epidemiologists, we can offer some lights with respect to clinical mechanisms. For example, it's possible that some of the effects on infant mortality are mediated through preterm birth, but certainly we cannot provide insights into the molecular mechanisms, and for that, some more crosstalk with the colleagues, I guess that like you would be extremely illuminating. We are also thinking on examining what the role is of grandparental exposures with longer-term clinical outcomes of the grand offspring, examining other exposures of the distal generation, like stress, and then hopefully being able to differentiate those intergenerational effects from true transgenerational effects. But for that, we probably need an F3 generation, and those studies will likely be conducted by the postdocs of our postdocs. All right, because by the way, these are the only sort of data sources that I am aware of where we could do these studies, and obviously it takes time for the populations to accrue pregnancies and data. And just very quickly, some acknowledgements. One to my friend and collaborator and really accomplice on these incursions, Professor Zvenkin, I think is from Karolinska Institute and the NIH for funding this exploratory work. Thank you. Questions?
Can I ask, in the case that you're showing from the grandmother and the mother in terms of obesity, the case is that the mother is also obese, or is mainly the mother is obese? Yeah. At the beginning, no, you have a difference. You studied the obesity and after each pregnancy. That's what I was confused with. In the model that you described,
is early pregnancy early pregnancy obesity?
It's early pregnancy obesity.
Yes. Okay. On average at week 10.
If the grandmother has increased in every pregnancy obesity, that has an impact also on the daughter that will follow the same pattern. So I know you look at the chart.
Yes. Yeah. So the answer to your question is yes. That was the first part of the research that we did almost 20 years ago. So yes.
There is a curve.
Yes.
Okay.
Absolutely.
And then you see now in the third generation what is now.
But not all of it is through the mother's obesity.
It's the grandmother's obesity.
Exactly.
So the F0 to F2 data, was the F2 data always taken from children of mother, or was it taken from children of father?
Aha. Great question. What I showed is only of children from a maternal grandmother, so from women.
Mother.
Yeah. We also have the data and some results for paternal grandmothers, right? But we cannot make sense of them. And I guess we are in the discipline of just not showing things because we can, but we really need to understand it. And even though we may have some hypotheses, really, they aren't very strong. So that's a bit of a conundrum for me because I just don't know what to do. But yeah.
Because it would mean then that the implication would be that the F1 generation are responsible for some of it, right?
Yeah. Fathers are also responsible. Right? Yes. How? I don't know. That's where I need the basic science.
We'll take one last.
Sure. Sorry. So just to add that in the US, even though we don't have like a great database as you have in the Scandinavian countries, Utah actually has a very good database called the Utah Intergenerational data. Each time, every year at SMFM, they get a plenary just because of that. It starts from the 1930s.
Oh, wow.
It's called the Mormons or something like that.
Yeah.
They have like great grandparents who they have complete data of. So if you
They have clinical data.
yes, they have everything. So and they even presented at SMFM and it's just amazing.
Yeah, that yeah. But we have BMI, for example.
They do have BMI. Okay. Yeah. But the question I wanted to ask is, did you look at type modification like by age or any other thing?
Age of whom?
Of the grandparents when they had
no, we have not. But what would you have in?
Because there's a direct association between age and BMI as well.
Oh, absolutely, absolutely. I mean, it's adjusted for age and height because it's a fact of life that weight accrues after each birth. But no, we didn't look at effect modification, much because of power. Because believe it or not, there were only about 900 stillbirths, even in such large population. So power becomes an issue. But
sure, absolutely. Excellent.
Thank you.
So we have come to the last seminar of the day, and I would like to introduce Dr. Kevin Theis, Associate Professor in Biochemistry, Microbiology, and Immunology at the Wayne State School, University School of Medicine. And the title of the talk is The Vaginal Micro-Immuno Environment and Preterm Birth. Thank you.
First, I want to thank you for the invitation to share the research of my collaborators and I on the potential roles of the microbiome and the immunoproteome on preterm birth. And to start, I want you to first think of some ways that you would characterize these individuals, some descriptive terms, some adjectives. You don't have to yell them out, just come up with a few in your head. You got some? Okay, so I'm guessing they're things like mother, child, parent, offspring, loving, tired, something like that.
Tired, I thought about the
tired. Okay. Did anyone have anything in their head come up in their head that has to do with the microbiome? Did anyone say that what's here is an amalgam, a consortium of host of microbial cells and alleles? Because that's what we are. And so you've all heard these numbers before over the last decade or so when you've sat in talks. You've heard that at least half of the cells and the vast majority of unique genes associated with your body are microbial in origin. And you've been told that this is important. And if you haven't, I'm telling you now, because your microbes are producing biomolecules at all times, every time they metabolize. And you're in constant intimate association with those biomolecules. And in fact, at some points in the day, a third of the metabolites circulating through your bloodstream are from your microbiome. So the most important aspect of this is that your phenotype is at least in part determined by your microbiome. So back in the early and mid 20th century, there was the nature versus nurture debate. The question was, is your phenotype due to your genome, or is it due to the environment in which you grew up or inhabit? And the ultimate solution to that was that all phenotypes are necessarily products of gene-environment interactions, and underlying variation in phenotypes is due to, variation in phenotypes is due to underlying variation in genomic profiles and environments. But one thing that I propose to you, and I hope you take away from this talk, is that we're missing a key element. And that is that we also need to include the genomes of the microbes that inhabit our body. And so the idea here is that when we look at an individual animal or plant, I suggest that they're best perceived as actually a holobiont or a hologenome. So in other words, they're not autonomous individuals. They're instead emergent entities that are products of these interactions between their genomes, their microbiomes, and their environments. The holobiont would simply be the host plus the microorganisms inhabiting it at any given time, and the hologenome would be the summary genomic content. So the question obviously is, okay, is this just some ivory tower concept? Why am I even bringing this up? And our ideas and our perceptions should have applied value just like our more deliverable scientific products. So I assert that it's not simply an ivory tower concept, and that it contributes directly to evolutionary theory. It complements what Darwin put forth. I don't have time, obviously, to get into this today. I'll just leave you with this idea. There has never been an animal or plant that competed for reproductive opportunities within its population without at least some aspect of its phenotype being determined by the interaction it has with its microbes. And so if you're simply going to study the evolution of animals and plants and you don't take the microbiome into account, I think you're missing a big part of the story. But we're not here to talk about evolution today. We're here to talk about more medical related issues. And so I also want to suggest that the concept of holobionts and hologenomes has immediate functional utility as well, particularly with respect to medicine. And so the focus of my lab is on, and my collaborators, is on the microbiome, the immunoproteome, and preterm birth. So as has been mentioned, preterm birth is the leading cause of neonatal morbidity and mortality worldwide. Current rates are at approximately 11%. That means that 11% of all newborn babies are coming into a situation where they experience a microbial assault before their bodies were actually prepared for them to have that experience. The rates of preterm birth can be higher in urban environments, including Detroit, Michigan, where we are, where the rate is 15.1%, the highest in the United States. Also, Detroit notably has a large African-American population, and this is a patient group that has a disproportionate extent of negative maternal-fetal health outcomes. And of course, that's one of the reasons that the perinatology research branch was here for the last couple of decades. Now, when we talk about preterm birth, it can be subclassified. The most simple definition is just that it's any birth that occurs before 37 weeks, but we can also break it down into early preterm birth, which we define as before 34 weeks, and late being 34 to 37 weeks. We can also further subclassify it into iatrogenic and spontaneous. Iatrogenic would be clinically identified or clinically indicated. So for example, preeclampsia. And then there's spontaneous, which can further be subclassified into preterm pre-labor rupture of membranes and spontaneous preterm labor with intact membranes. And the focus of the rest of my talk is simply on spontaneous preterm birth. So spontaneous preterm birth is a syndrome. It has multiple underlying etiologies. One that has been studied in quite a bit and has been experimentally demonstrated is intraamniotic infection. Now, I do want to point out that typically the mammalian amniotic cavity is sterile. And so in a series of experiments using humans, non-human primates, and mice, we've demonstrated this over the last half decade. And so the question becomes, okay, well, when there are microbes within the amniotic cavity, where are they typically coming from? And the answer is that they typically come from the vagina, ascending through the cervix into the amniotic cavity. So that leads to the big question of, does the structure of the vaginal microbiome early in pregnancy predict whether or not a woman is going to ultimately exhibit preterm birth? And so this is a question that's been asked a lot. And I'm going to tell you a bit about that. But first, I just want to tell you about the structure of the vaginal microbiome. So if you look worldwide at reproductive age women, they're going to exhibit one of these in general. They're going to exhibit one of these 5 community state types. 4 of them are dominated by lactobacillus species, whether that be crispatus, Jensenii, or Gisseri, inners, or Jensenii. And then there's this other community state type that we call community state type 4. In this instance, the vaginal microbiome is not dominated by any one lactobacillus species. Instead, it's a more alpha diverse community populated by anaerobes such as Adipovium, Mobiluncus, Sneathia, Gardnerella, Prevotella, etc. And that's important because when women do have intraamniotic infection, it's typically those microbes that are part of community state type 4, as well as the genital mycoplasmas. It's not the lactobacillus dominating community, lactobacillus species. All right, so as I mentioned, this is a big question that's been of interest to many. And so over the last decade or so, there's been dozens of studies that have used molecular tools to investigate if the vaginal microbiome is predictive of preterm birth. And I want to point out that most of these studies have been conducted on the eastern half of the United States, in Great Britain, and in Eastern Asia. And there's this big gap of studies on the African continent where the issue of preterm birth is obviously quite substantial. And so I see this as one potential area where Wayne State can make some inroads. All right, so all of these studies have been done, but the issue is that the results from them are contradictory. There are studies that indicate that there's no association between the vaginal microbiome and preterm birth. Others that indicate it all depends upon the number of bacterial species present. Another one that says, okay, if community state type 4 is present early, then preterm birth is likely. Others say, no, it's not community state type 4, but it's specific microbes that are typically found within community state type 4. And so the issue is, okay, all those studies, contradictory results. Well, what's the source of the contradiction? And I'll propose a couple here. One is sample sizes. The studies had small sample sizes, which leads to low power. Another is ethnicity. Ethnicity influences the structure of the vaginal microbiome. So if ethnicity cannot be controlled for within a study, then that could introduce noise. Another one is subcategorizing preterm birth. A lot of these studies just studied preterm birth, delivery before 37 weeks. They didn't subcategorize it as I just proposed. Another is strain level analysis. So I just told you about Lactobacillus crispatus and inners, et cetera. But this phenomenon may actually be occurring, if there is a causal role of the microbiome, it may be occurring at a level less than the species. It may be at a strain level. As you know, bacteria are very good at horizontal gene transfer and sharing pathogenicity islands. So we have to operate at a more fine resolution. And the last one is, these studies typically did not take into account the host immune response to the microbiome that's present. And we know that disease is not defined by microorganisms themselves. It's defined by how the host responds to a potential microbial assault. All right, so this work that I'm about to share with you was conducted by a multidisciplinary team within the perinatology research branch here at Wayne State University. And in particular, I was the head of the microbiome unit. Dr. Tarka was the head of the computational biology unit. Dr. Gomez-Lopez was the head of the immunobiology unit. And obviously, Dr. Romero was the branch chief who oversaw the execution of all of these projects. These are other people who contributed directly to the research that I'm presenting here. And I just want to point out that these samples took decades to collect. That's an important point there. Here are people who contributed in a very substantial way to these projects. Andrew Winters and Violetta Florova were postdocs in my unit. Jonathan Panzer just completed his Ph.D. recently, and Zachary Schaefer is an MD Ph.D. student in the lab. All right, so the study design was that we collected vaginal fluid longitudinally across pregnancy from 8 weeks up to 37 weeks from 771 women. 257 of those women delivered preterm, 148 with spontaneous preterm labor with intact membranes, and 109 with PPROM, preterm pre-labor rupture membranes. All of the women contributed at least 3 vaginal fluid samples. Almost all contributed 4. That resulted in 3,000 samples, which constitutes the largest study conducted to date. Notably, we stopped sampling at 37 weeks. So that's been another potential issue with some of the other studies, is the samples representing the term delivery group were actually collected after the period at which all of the preterm individuals would have delivered. And so we don't have that issue here. Some of the clinical and demographic characteristics of note, just to repeat, this is a largely African-American population. And has been discussed a number of times today, you would predict that the women who ultimately delivered preterm were the ones who had a history of prior preterm birth. So to characterize the microbiome, we did 16S ribosomal RNA gene sequencing, as well as metagenomic sequencing. This allowed us to characterize the alpha diversity of the microbiome, as well as to get taxonomic profiles. We also characterized the community state type. And since we did metagenomics, we were able to get some information on functional pathways and potential virulence factors. But ultimately, the end game was, could the structure of the microbiome early in pregnancy predict the likelihood of a woman experiencing spontaneous preterm birth? So these papers are all either published or available as preprints. All right, so then with respect to the immunoproteome, we characterized the chemokines, we characterized the cytokines, and we categorized antimicrobial peptides. And again, then we asked, how well does the immunoproteome predict the likelihood of preterm birth? And I'm going to focus primarily on the microbiome data. I'll show you some of the results on the immunoproteome, but I'm not going to go into too much detail on that. And again, this is available as a preprint as well. All right, so what is the structure of the vaginal microbiome across pregnancy resulting in term birth? So here is another heat map, and you can see on the top that it's, again, community state types 1 through 5. The only difference here is that community state type 4 has been divided into community state type 4A and B. The only difference there is that community state type A is more has a higher relative abundance of Candidata selachnicurva vaginae, and B has more Gardnerella and Adipobium. But again, you see these same community state types that were in the original heat map that I showed you. So women in Detroit have the same microbiomes as women worldwide, and women who are pregnant have the same microbiomes as those who are not pregnant. They're just different in relative abundances amongst those different groups. One of the things that I want to point out here, I find this fascinating at how predictable the vaginal microbiome is amongst reproductive age women worldwide. When we first started talking about holobionts and hologenomes, one of the principal criticisms was, well, the microbiome is incredibly diverse, and it's not really it's so diverse it can't really be heritable. So what value is there really in thinking about the microbiome in any kind of evolutionary context of the host? But I think that perception comes from people focusing on the gut microbiome, which is incredibly diverse. But I just showed you that the vaginal microbiome is predictable. There are studies coming out now that it's potentially heritable, either through vaginal birth or later on in life through social contact, post-puberty. And then it can factor into reproductive success, whether that is the success of fertilization, pregnancy outcome, or the seeding of the neonate, which is going to be very important in priming the neonate's immune system. All right, so across gestation, alpha diversity decreases. So that means two things. One, as gestation progresses, you have fewer different species within the vaginal microbiome, and also those that are present are more evenly distributed. It's not dominated by one particular bacterium like a lactobacillus per se. So here's what's actually going on. The vaginal microbiomes that were originally community state type 4 early in gestation, those are transitioning to community state types 1 and 3. One's dominated by Lactobacillus crispatus and Lactobacillus inners. Of course, if you have a reduction in community state type 4, then you're going to have a reduction in some of the typical members of community state type 4. And so just to give you an idea here, here's Lactobacillus. Its relative abundance increases across gestation. And then these different members of community state type 4, Smithia, Adipobium, Fastidiosipila, Megaspira, Gardnerella, Prevotella, those are the ones that are decreasing across typical gestation. All right, but does the structure of the vaginal microbiome vary with birth outcome? And remember, these are our groups, those that delivered a term, those that had preterm labor with intact membranes, and those that experienced P product. Here is a heat map again, and what I want you to take away from this is that community state type in and of itself is not predicting the likelihood of whether or not a woman ultimately delivers preterm. So a woman can have the community state type dominated by lactobacillus crispus and still ultimately deliver preterm, and a woman could have community state type 4 and deliver a term. So that's too broad a way to look at these data. But one thing that was predictive was alpha diversity. So here you can see that individuals who delivered preterm tended to have more alpha diverse microbiomes than did those that delivered a term. If you then break down the spontaneous preterm delivery into PTL and PPROM, you see that it's actually the women who experience PPROM that have the higher alpha diverse microbiomes early in pregnancy. And ultimately, if you break it down further, it's specifically those women who exhibit early PPROM. That is PPROM that occurs before 34 weeks. What's going on here is that this is the pattern from the controls. This is when I showed you where there's a shift from community state types 4 to 1 and 3. You see that amongst women who exhibit PTL. You see that amongst women who exhibit PPROM. But where you don't see it is women who ultimately exhibit early PPROM. When they have community state type 4, it persists. And when they have community state type 1 or 3, it can even sometimes transition to community state type 4 as pregnancy progresses. Again, this is speaking at the community state type level. If we look at microbes typical of that community state type, it was specifically Anaerococcus, Mobiluncus, Prevotella, and Sneathia that seem to be most associated with cases of early PPROM. And this is particularly interesting because Sneathia has the capacity, when it transcends the cervix, to jeopardize the integrity of the fetal membranes. That's been demonstrated in vitro. So, predictability of the vaginal microbiome, notably using 16S ribosomal RNA gene data. So this would be characterizing in kind of a crass way the vaginal microbiome. You can see that with respect to spontaneous preterm birth in general, no predictability, also no predictability for PTL or for PPROM in general, but PPROM less than 34 weeks, you have some degree of predictability, but notice that the AUC is very low. So modest predictability at best. My takeaway here, this is the same thing. It's just on the top is individuals who ultimately experience PTL, and then the bottom PPROM, and you can see that there's a relationship between a diverse microbiome in early pregnancy and the gestational age at delivery. However, it's again a modest association. And so my takeaway from this would be that using the vaginal microbiome in early pregnancy, if you're going to characterize it using the 16S gene, and in particular just a small snippet of the 16S gene, you're not going to get the resolution required to get any kind of predictive value of incorporating this into clinical use at this particular time. Okay, but that's 16S. How about metagenomics? Are the results similar or dissimilar? Well, actually, all of these samples were sequenced using 16S, ribosomal RNA gene, and metagenomic sequencing. And if you look at this line that's right in the middle, that line means that if a microbe falls on that line, it was equally represented using both types of sequencing. And it's actually remarkable how well they did. There's only 2 microbes that seem to have been biased using the 2 approaches. One is Lactobacillus iners, and one is Gardnerella. So now, if you're studying something like bacterial vaginosis, then you need to take this into account. But I think if you're doing a general vaginal microbiome study, 16S is going to essentially give you the same answer as metagenomics, just without the metabolic data, but a more cost-effective approach. So given that the types were so similar, the results using the 2 approaches were also very similar. So I'm just going to give you a highlight. Variation in some particular strains across birth outcomes, and the focus here is on early PPROM. So these are ultimately the microbes listed in order of importance and how they contributed to predictability. And then we additionally ran a linear discriminant analysis of effect size to say, okay, which microbes are specifically associated with early PPROM. And what we come up with is Sneathia, Prevotella, GBS, Gardnerella are the main ones. So again, Sneathia and Prevotella and Gardnerella keep coming up. And again, all of those are typical members of community state type 4, but community state type 4 in and of itself is not predictive. Okay, so how about at a functional level? So this is a volcano plot, and in the upper left, you can see that there were 4 pathways that were enriched in term controls, and in the upper right, 2 that were enriched in cases of early PPROM. The ones that were enriched in term controls were for nucleotide biosynthesis, and the ones that were enriched in early PPROM are for carbohydrate biosynthesis and metabolism. If you look at which bacterial types were, that is genomic species, if you will, were responsible for these, then the Calvin pathway was attributed to Prevotella and Sneathia, and the pathways dealing with nucleotide biosynthesis were attributed to Lactobacillus inners and Lactobacillus crispatus. So what's my takeaway from this? Unfortunately, I think this is a reflection of the underlying taxonomy. And so what's happening is those were the microbes that were associated with those controls and cases. And so these particular pathways are simply associated with them. However, I think that what this also more importantly points to is that we need to increase the robustness of our functional databases, particularly as they pertain to the vaginal microbiome, because we're clearly not getting the data that we deserve to get, given how often the vaginal microbiome is studied. So I think a lot of work needs to put into that, and that's going to require culture, which I'll talk about right at the end. Okay, so predictability of the vaginal microbiome for preterm birth was, again, not good, except if you focus on early PPROM in particular. And here you can see that our AUC is now up to 0.72. So if we do get more resolution, maybe that can start to go up. But how about the immunoproteome, the host response to any potential microbial community that's there? And what we show here, I'm just showing you the predictive aspect of the immunoproteome data. Spontaneous preterm birth, not predicted. Spontaneous preterm labor, not predicted. Spontaneous preterm labor, less than 34 weeks, not predicted. PPROM overall, not predicted. But early PPROM, that's predicted, and actually with quite good AUC levels. So what this is telling us, so the black line is the vaginal immunoproteome data. The blue line are maternal characteristics, including maternal age, parity, prior history of preterm birth, and BMI. So the immunoproteome data are far more predictive than those maternal characteristics. And if you add the maternal characteristics to the immunoproteome data, it's not improving the quality of the model. So what are the conclusions? The vaginal microbiome in and of itself is not predictive of spontaneous preterm birth in a general sense. However, aspects of the vaginal microbiome and the vaginal immunoproteome can be predictive of a particular subset of spontaneous preterm birth, notably early PPROM. And the characteristics of those would be that the vaginal microbiome early in pregnancy is typically going to be more rich, have more community state type 4, have more representation of community state type 4 particular microbes. The vaginal fluid will be more pro-inflammatory, including IL-6 and IL-1 beta, and will have reduced antimicrobial peptides, particularly beta defensin 2. But so it may be a little bit of a disappointment that these weren't predictive of spontaneous preterm birth overall. But think about early PPROM. Obviously, the mechanisms behind PPROM are going to be different than those behind spontaneous preterm labor with intact membranes. And cases of early PPROM suffer pretty adverse perinatal outcomes, including death. And it would make sense that PPROM, which is the rupture of membranes, is perhaps going to be most reflective of the structure of the vaginal microbiome, which could potentially ascend, compromise the integrity of the membranes and lead to PPROM, or actually be affected by PPROM after the membranes are ruptured and there's amniotic fluid and vaginal fluid exchange. So future directions. These are what we've accomplished so far with this data set. The next thing is to do a strain level analysis. So even what I just showed you was metagenomic assigned species. but we now need to do metagenomic assembled genomes and actually look at this at the strain level, because a single species can have multiple different strains with different virulence capacity. The other thing is we haven't looked at whether you combine the micro and immuno data, does that increase the predictability? The other thing is that when we do metagenomic sequencing, we have data on the host genome, but we haven't incorporated that. So if we're going to take a whole genomic perspective, then what we really need to do is incorporate information on the host genome, the microbiome, and the immunoproteome early in pregnancy, as whether or not it's predictive of spontaneous preterm birth. And then the last thing, as I mentioned, to help improve the database, but also to allow us to do experimental work in the laboratory in vitro or animal models, we need to get some of these key players in the vaginal microbiome into consistent and reliable culture, which right now we can't do. There's some key members where we only know about them because of the molecular data that we have of them. So I think effort needs to be invested in improving those culture libraries. Thank you.
Do we know if vaginal progesterone affects the microbiome?
The studies that are out are ambiguous in that. There's some contradiction there. I can tell you that unpublished data that we have does not suggest that it does. The vehicle and progesterone yielded the same structured microbiome post-administration, but it's not published.
So given that you want to increase your resolution, do you have sufficient sequencing data to do that? What in hand?
So I do. The only thing I'm worried about is potential depth. I know that I can get good assembled genomes of the principal members, but some of the other ones, it's going to be more difficult. Like the general mycoplasmas, which I didn't talk about that much here, they're actually, if a woman has intraamniotic infection, those are the ones that are most often present, yet they come up at very low relative abundances in any sequencing survey. They're present, but they're not there at high relative abundance. So to answer your question, for some of the key players, yes, I think I have the data in hand to do the resolution right now. Just need postdocs.
just as follow-up. So I'm kind of curious that you left the other half of the equation out, and that's all these microbes.
So I haven't totally left it out. I mean, I've thought about it when I'm having discussions with colleagues, but yes, we did not, at the perinatology research branch, we did not sample any penile swabs of fathers. So I don't have those data. Marta.
Very nice. So how different is the microbiome during early pregnancy from the pre-pregnancy microbiome and the after pregnancy microbiome? And will those be used to predict the success or the early preterm birth?
So that's a very good question, and it's a slightly complicated one, but pre-pregnancy and early pregnancy, the literature suggests not too different. The actual act of mating can change the microbiome as well, though, so you can get temporary shifts. I showed you that the microbiome clearly changes across gestation and shifts toward these lactobacillus dominant. One of the reasons I see it so clearly is the population that we work with. You see that very pronounced in populations of women that are African American or from Africa. You don't necessarily see that in other populations of women. Then in terms of, so what I showed is specific to my population. In terms of post-pregnancy, there is a pronounced shift. So you can have a lactobacillus dominated community right as you're going up to labor. When labor starts, I think the current data suggests that it's the process of labor itself that's changing it, but you could think that delivery is going to have an effect too. What you find is a more typical community state type 4 community that can persist for 6 months even. So there's definitely a change in the microbiome with delivery.
Can you really, really nail it down? What would you do about it?
So that's a very good question. I think that if we could get to the point, there's new technology called nanopore, where within 10 to 12 minutes, you could go ahead and know the vaginal microbiome of a woman when she comes in. And nanopore gives you very long bases. So it's not like what I just showed you was a 250 bases. Here you could sequence the entire 16S gene. You could get strain level identification right when the woman's even in the office. So then what do you do? Well, it's going to be prebiotics, probiotics, or a symbiotic approach. I think it's going to have to be some kind of Lactobacillus crispatus type microorganism that can persist in the vagina, can compete, and that is shown to be safe. But then I think you're also going to have to give some kind of prebiotic to help that probiotic do well and outcompete, and that's what I mean by symbiotic. Now that may also depend on the woman, so that might have to ultimately be a personalized approach. And this would just be another example of personalized microbiome intervention. But I think if there is going to be one place where prebiotics and probiotics may be of value, it is in something like this, if the microbiome is truly a cause of preterm birth, which, as I showed, it's a syndrome that has many, many causes. So you'd have to be really sure that the change you're making is important.
How close to pre-prom, that early pre-prom, is the sampling?
No sample was collected after rupture of membranes.
No, but how close prior to rupture of membranes?
You know, I want to say 2 weeks, but I don't remember exactly.
Well, we can have any range, from a few days to a few weeks. The data we have shown, for instance, up to 28 weeks. So you consider the data only up to 28 weeks, and you want to predict something that happens after 28 weeks. Otherwise, what you're seeing is just an imbalance in the gestational age sampling, what most people do, and they find differences in the microbiome predicting the preterm birth, but it's actually the association between the time of gestation and the diversity that is the two. So we have a span, but does not affect the hour.
So my question is, though, is if it's fairly close to when pre-prom is occurring, is the data you're seeing reflective of the infection that's in the amniotic cavity, and that's what, and it's not a criticism, it's actually, can you use the vaginal microbiome to define whether or not a woman is infected? It's much easier than an IL-6.
So I don't know the answer to that last part, but that's definitely something to consider. But I can tell you that Dr. Gomez-Lopez has just published a paper last week where they show that when you cause intraamniotic infection through multiple means, even with intact membranes, it changes the structure of the microbiome in a mouse.
So it works both ways.
It works both ways.
Take one last question. Yeah. Thank you for the education. I always get interested in microbiomes, but I think you made me really understand it. Very interesting to see that the Jack Ravel's paper is still cited over 4,000 times, or the highest so far in medicine. But my question is, when you guys collect this in pregnancy, do you use speculums that lubricate? And if so, don't you think that the lubricants we use to please those speculums can affect the microbiome?
Do I have any of those?
We're not lubricated.
Okay.
They were not.
Yeah.
It's definitely all speculum guided collection, but thank you.
Yeah, thank you. Yeah.
Thank you. The talks during the symposium have spanned an entire breadth of pregnancy. We covered implantation, fetal development, all the way to delivery. And the talks covered the etiology of preterm birth. We spoke about it from the microbiome, HIV infection, reproductive immunology, gestational exposures, omics technologies, and all this we did. The talks covered from a basic science perspective, from clinical, translational, implementation science, intergenerational epidemiology, and we also covered postpartum depression prevention and maternal health equity. I've never seen a symposium where all the speakers were just on time. I really want to thank all the speakers because you all have such a busy schedule that you still took some time from that busy schedule and you are here with us at the Mott Center at Detroit. And this symposium has brought together renowned scientists from the major research institutions in the state of Michigan, Michigan State University, University of Michigan, and the Wayne State University, and in the field of maternal-fetal health. And I would like to also thank members of the Mott Center who have really helped us to do the symposium. Sharon, Alyssa, George, Alex from my lab, Joe, Hong, Sapna, and Alex from Dr. Moore's lab, and Melissa and Nick. And if I missed any names, I apologize. This symposium is the vision of our Center Director, Dr. Villamor, and who's a renowned reproductive immunologist. And it would not have happened but for his enthusiasm and vision, and for supporting all the members here at the back. I would like to invite Dr. Moore to say any concluding words and also about the evening panel discussion. Thank you.
I just joined again. Thank you, everybody, for participating in this. We try to make it as more variable so everybody learns a little from it. I learn a lot. I always learn a lot. And I think that is the objective. Even many of the areas is not my direct area of interest, but if we don't learn other things, we always be just focused in a little hole, in a little box, and we never think out of the box. I say I hope the students, and I hope the students should be, all the students should have it here. We know we have people in the Zoom, so thank you to all the speakers, and thank you, Jay, for organizing. Now, our mission is the community. So we don't just teach out between ourselves. If what we learn here, it doesn't go out, we are missing the main point of what we get the money from the taxpayers. So that is the reason of after educating ourselves and sharing the knowledge that you have in the evening, in a couple of hours, around 6 o'clock, in an hour, we will open through Zoom to all the public in the community, what we call the panel of experts. So if the speakers are able to stay, we will really appreciate, because we will open and people will ask questions. Yeah, I will modulate the discussion, but it's mainly to answer questions from patients who, when they see their doctors, they don't have the time to answer their questions. Yeah. And they want to know so many questions, so many things. So that will be the objective of 6 o'clock. Yeah, it will be a panel of discussion open for the public, and we will see what it comes. There's always an unknown experience. We don't know what type of questions we will have. Yeah. So again, thank you very much, and to the speakers, I'll see you a little before 6 o'clock. Thank you.
The Zoom information for the 6 p.m. session has already been shared. We ask that the speakers please stay here.
It's in this room. We will come back to Zoom. Now, if you want to share with anybody, please feel free to share the Zoom.
Yeah, I think so maybe just on it if you are a social team.